Methods within the framework of technical processes in industrial production. It involves the application of microbiology and biochemistry together with technical chemistry and process engineering. 4. The definition provided by the Food and Drug Administration (FDA) of the United States of America describes biotechnology as the application of biological systems and organisms to technical and industrial processes. 5. Organization for Economic Cooperation and Development (OECD) proposed that biotechnology be described as the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. This definition is regarded as well informed and acceptable. However, in a more complete sense, biotechnology can be defined as the application of science and technology to living organisms, as well as parts, products, and models thereof, to alter living or non-living materials for the production of knowledge, goods, and services. Disciplines of biotechnology. Almost every branch of modern biology shares its contribution to the development of biotechnology as a subject. As an interdisciplinary field, biotechnology is a composite of branches of basic and applied life science as well as a few branches of engineering science. Multiple disciplines such as cell biology, genetics, biochemistry, molecular biology, microbiology, plant and animal biology, genomics, proteomics, computational biology, bioinformatics, developments in bioprocess technology, genetic engineering, protein engineering, chemical engineering, and mechanical engineering, food technology, or NCLUDI, in the area of biotechnology. 12. Applied biology. Advanced biotechnology. Biotechnology, an overview. Biotechnology, in a broader sense, describes a discipline consisting of bio, related to biological organisms, processes, and associated relevance, and technology related to the application of scientific knowledge to the practical aims of human life. As a technology, it is as old as human civilization. Since the human civilization, it has been used for multiple purposes such as plant and animal breeding, fermentation for producing curd, bread, and beer. However, the emergence of the subject as a branch of science is relatively new. It began with the emergence and advancement of other branches of biological science such as cell biology, genetics, microbiology, biochemistry, and molecular biology. The term biotechnology was first used in 1919 by Hungarian engineer Karoly Ereky in his book, Biotechnology of Meat, Fat and Milk Production in an Agricultural Large-Scale Farm. However, Karoly Ereky's concept of biotechnology was relatively simple compared to the modern-day definition of biotechnology. It denotes biotechnology as a bioprocessing method that helps to produce useful consumables. The definition of biotechnology has been vividly changed since that time. Interestingly, there are more than one definitions of biotechnology, depending on the country and organization. Some of these are as follows. 1. In Japan, biotechnology is described as a technology using biological phenomena for copying and manufacturing various kinds of useful substances. 2. In France, a working definition is biotechnology consists of the industrial exploitation of the potential of microorganisms, animal and plant cells, and subcellular fractions derived from them. 3. In Germany, it is said that biotechnology deals with the introduction of biological as well as other drugs used in the biotechnology sector. An example includes the production of insulin hormone from the genetically modified E. coli. It also contributes to the development of diagnostic tools and techniques relevant to human health and diseases. The emergence of stem cell therapy for treating genetic disorders is another significant contribution to this area of biotechnology. Green biotechnology. This discipline of biotechnology covers agriculture and related area. Primarily, it aims to develop transgenic crops and more plants with better yield. Introduction of a foreign gene into the plant genome often results in a better variety that can survive certain environmental challenges. Additionally, an eco-friendly practice of agriculture using these plants might reduce the use of pesticides, and this can be counted as a great benefit of green biotechnology. Blue biotechnology. This is a relatively less popular area that deals with marine organisms as well as related health and the environment. Marine pisciculture and aquaculture, biofilms, biological oceanography, and bioremediation are the broader areas that come under this discipline. Emergence of molecular biotechnology. Molecular biotechnology deals with the usage of various laboratory techniques to manipulate DNA and subsequently the protein in order to modify an organism for the benefit of mankind. Actually, molecular biotechnology forms the basic groundwork of almost all modern-day biotechnological applications. This field has emerged as a result of the coevolution of many other related subjects such as molecular biology, genetics, cell biology, and biochemistry. It is difficult to pinpoint the exact time point of the emergence of molecular biotechnology. However, the discovery of DNA double helix by Watson and Crick in 1953 can be considered an important landmark in the history of the emergence of molecular biotechnology. Some other contemporary as well as subsequent discoveries also immensely contributed to the emergence of this specific. Figure 12.1. Disciplines of biotechnology. A number of basic and applied fields of biology, such as cell and molecular biology, genetics, and biochemistry, along with other fields of engineering constitute the subject biotechnology. Many other branches of science, such as computer science, statistics, and bioinformatics significantly contribute to the development of the different disciplines of biotechnology. However, according to the sector of usage, biotechnology has been divided into 4 color-coded disciplines, namely white biotechnology, red biotechnology, green biotechnology, and blue biotechnology. Below, we will have a brief overview of these different disciplines. White biotechnology. It refers to the sector of industrial biotechnology. This branch deals with the manufacturing and development of biofriendly products such as genetically modified organisms, biofuel, and biopolymers plastic at the industrial level. These products are mostly used by other related industries for downstream processes. For example, genetically modified bacteria, Ralstonia eutropha, is grown at an industrial level for the subsequent production of poly-3-hydroxybutyrate (PHB), a bioplastic. Cost-effectiveness and eco-friendly nature are two major advantages of this field. Red biotechnology. It indicates the field of medical biotechnology. It is involved in the production and improvement of antibiotics as bond. Chronologically, the development of this field started in the early 1970s by a group of scientists who started experimenting on bacterial plasmids, the extrachromosomal circular DNA that is capable of self-replicating along with the bacterial DNA. Subsequent discovery of restriction endonucleases for cutting the plasmids at a specific site stimulated the effort to make novel plasmids with foreign DNA, which can be propagated into a host bacterium such as E. coli. In the first decade of 1970, the pioneering groups of scientists namely Paul Berg, Stanley Cohen, and Herbert Boyer successfully produced the first rDNA molecule in the laboratory. In 1972, Paul Berg, a recipient of the Nobel Prize, made recombinant DNA for the first time in a lab. He used the DNA from 2 viruses, namely lambda phage and simian virus SV40, performed restriction digestion and ligated 2 pieces of DNA to make an rDNA molecule. However, he did not put the rDNA into a bacterial host. Area of biotechnology. One of the major discoveries that tremendously influenced the emergence of molecular biotechnology was the finding of extrachromosomal plasmids in bacteria. The observation of extrachromosomal DNA in bacteria was reported in the 1940s while describing the mechanisms of antibiotic resistance in bacteria. Later on, in 1952, Joshua Lederberg put forward a working definition of these extrachromosomal DNA as plasmid. Another event that significantly influenced the progress of this field was the discovery of restriction endonucleases in bacteria as viral defense machinery in the 1960s. These restriction endonuclease enzymes can cut the double-stranded DNA at specific sites. Additionally, the discovery of DNA ligases in 1967 by Gellert, Lehman, Richardson, and Hurwitz played a key role in the emergence of molecular biotechnology. DNA ligases are enzymes that can join 2 pieces of DNA by catalyzing the formation of a phosphodiester. AB. Figure 12.28. Production of rDNA molecule. B. Professor Paul Berg. Originally, the vector DNA from SV40 virus and the DNA from bacterial lambda virus were cut using the E-coli restriction enzyme to produce the same overhang in DNA. These 2 pieces of DNA were ligated to produce the chimeric DNA molecule. This approach was first adopted by Paul Berg in 1972. In 1973, Boyer and Cohen made recombinant DNA using a condomycin-resistant plasmid and a tetracycline-resistant plasmid. Subsequently led to many other useful discoveries. For example, hybridomas were created for the first time in 1975 and the monoclonal antibodies and human insulin produced in bacteria were produced in 1982. They inserted this chimeric molecule into the E. coli and made the first genetically modified organism. Together, these groundbreaking inventions made the foundation of application-oriented molecular biotechnology, which ABC. Figure 12.38. Transformation of a bacterium with rDNA. Bacterial transformation is the successful introduction of a DNA molecule into the bacterial cell. In 1973, Boyer and Cohen produced rDNA and transformed the bacterial cells using the rDNA molecule. Thus, the credit for making the first genetically modified organism goes to these 2 scientists, B. Herbert Boyer and C. Stanley Cohen. 2 other scientific breakthroughs significantly influenced the development of this particular field. These were the discovery of DNA polymerase in 1956 by Arthur Kornberg and the discovery of polymerase chain reaction (PCR) by Karen Willis in 1985. Some of the major discoveries that led to the emergence of molecular biotechnology and the subsequent development of this field can be found in the table below. Event S. Year name of the scientist S. Discovery of extrachromosomal DNA as plasmid. 1952 Joshua Lederberg. Proposed DNA double helix model 1953 James D. Watson and Francis Crick. Discovery of DNA polymerase 1956 Arthur Kornberg. Discovery of genetic code 1961 M. W. Nirenberg and H. G. Grana. Discovery of DNA ligases 1967 Gellert, Lehman, Richardson, and Hurwitz. Produced the first rDNA molecule by recombinant lambda phage DNA and simian virus 40 DNA. 1972 Paul Berg. Event S. Year name of the scientist S. Produced the first genetically modified bacteria by introducing antibiotic resistance genes in E. coli. 1973 Stanley Cohen and Herbert Boyer. Produced the first genetically modified animal. A transgenic mouse was produced by inserting foreign DNA into its genome at the embryonic stage. 1974 Rudolf Janisch. Discovery of polymerase chain reaction (PCR). 1985 Carey Mullis. Table 12.1 Scientists with their contributions to molecular biotechnology. Recombinant DNA technology. Recombinant DNA (rDNA) technology is a science that deals with cutting and joining DNA fragments to produce a new combination of a functional DNA molecule in a laboratory setup. Invention of the technology transformed the field of traditional conventional biotechnology into application-oriented modern biotechnology. Recombinant DNA technology, which can also be considered as an art of cutting and pasting DNA pieces, includes a variety of tools and techniques to produce a new combination of DNA for a number of applications. Basically, a piece of DNA is cut using a restriction endonuclease enzyme and ligated into a pre-cut plasmid vector obtained from bacteria. This chimeric DNA molecule is recombinant DNA. However, just producing a piece of recombinant DNA does not make it fit for subsequent applications. For that, the rDNA needs to be inserted into a suitable host organism, preferably a bacterium. As the bacteria would divide multiple times in a proper growth condition, the rDNA molecule can be produced in many copies. Altogether, these processes are also known as gene cloning. The basic steps used in rDNA technology for the purpose of gene cloning are described below. 1. DNA isolation, extraction from an intended organism. 2. Restriction digestion, cutting the DNA in the cloning vector, where the DNA would be inserted with suitable restriction endonuclease enzymes. Cloning vector essentially contains three features. First, it would have an ORIC, a site that makes it capable of replication. Secondly, a marker gene, which is helpful for selecting the transformed host with this vector. Third is a multiple cloning site, MCS, which contains sites for multiple restriction enzymes. Restriction endonucleases are the enzymes that recognize and cut a specific sequence, usually 4 to 6 base pairs, of DNA. As a result of restriction digestion, the DNA produces either a double-stranded blunt end or a single-stranded sticky end. Depending on the strategy of cloning, the DNA and the vector are treated with specific sets of restriction enzymes. 3. Ligation, ligating the DNA in the cloning vector, which were digested with restriction endonuclease in the previous step, using the enzyme called ligase. Ligase joins two DNA molecules by catalyzing the formation of a phosphodiester bond between two DNA strands. This would produce the recombinant DNA that can be used in a subsequent transformation step. 4. Transformation, inserting the recombinant DNA vector plus desired DNA sequence molecule into a suitable host cell. Within a host, for example, bacteria, the rDNA could replicate to produce multiple copies to yield a large number of rDNA molecules. 5. Propagation of host cells. Successfully transformed organism, most commonly bacteria, needs to be cultured successfully in order to obtain large quantities of the recombinant DNA. Figure 12.4 Different steps of rDNA technology. The DNA to be cloned in a suitable vector are digested using restriction endonucleases. After this, the two pieces of digested DNA are ligated to produce a piece of recombinant DNA, which consists of both the vector and the desired piece of DNA. Then, an organism, for example, a bacterium, is transformed using the vector containing the desired DNA. After successful transformation, the bacteria are grown in large numbers using proper culture media. These transformed bacteria are subsequently used for different purposes. There are various applications of rDNA and associated technologies. For example, the genome of an organism contains multiple randomly distributed restriction sites, thus producing a unique pattern when treated with a combination of restriction endonucleases. This is known as restriction fragment length polymorphism (RFLP). This can be used for multiple purposes such as genome mapping, forensic tests, and the diagnosis of genetic diseases. Another example includes in vitro mutagenesis and cloning to study the function of a gene. Recombinant DNA technology can also be used for numerous applications for producing genetically modified organisms. Some of these applications can be found in the subsequent section. Application of molecular biotechnology in plant, animal, and agricultural sciences. Biotechnological advancement has propelled the field of plant science and its application in agriculture. Genetically modified plant in crop varieties with better yield as well as resistance against biotic and abiotic stress are produced. It has helped to develop better quality crops with a shorter lifespan and increased sustainability in different climate conditions. It is also claimed that some of these genetically modified agricultural products are enriched in food value. Similarly, the application of genetic engineering along with proper breeding methods has produced a better variety of animals. Higher quantity of milk-producing farm livestock and poultry birds with better yields of eggs and meat have been produced using animal biotechnological approaches. Altogether, it immensely helps to increase the outcome of agricultural output, thereby supplying an ample amount of food production. Also, using molecular biotechnology, transgenic organisms including plants and animals are produced, and these animals are immensely helpful for scientific research to gain insights into disease mechanisms and other biological processes. Figure 12.5 Steps of making transgenic mouse. For producing transgenic organisms, a number of approaches are taken. For example, to produce transgenic mice, transfected embryonic stem cells are cultured and selected for the subset expressing the desired gene. Then, the transformed cells are delivered into the inner cell mass of a cultured embryo and implanted in the uterus. Another method includes microinjection of the rDNA with the desired gene into the fertilized egg followed by uterine implantation. Finally, the offspring are tested for the presence of the desired gene in the selected heterozygous mice are crossed to produce homozygous strain of transgenic mice. In medicine, pharmaceutical sciences, and human health, identification, production of antibiotics and other drugs are a huge contribution of molecular biotechnology. Biopharma, a branch of molecular biotechnology, aims to develop pharmaceutical products using genomics and genetic engineering tools. Some of the major types of biopharmaceuticals comprise of hormones, blood coagulation factor, interferons, etc. More than half of these biopharmaceutical products are aiming to combat cancer. A significant number of drug-like molecules that were made using the tools and techniques of molecular biotechnology are under clinical trial. Additionally, the production of vaccines, antibodies for immunotherapy and diagnostic usage are also made using molecular biotechnology. Figure 12.6 Biotechnology helps in the production of drugs. For production of the drugs, the organisms are cultured in batch fermenters to grow them in large numbers. Then, using the help of biotechnology, compounds with medicinal values are extracted and purified. The above diagram shows the basic steps involved in the production of the antibiotic penicillin from the fungus Penicillium sp. Gene therapy and genetic counseling are also a contribution of biotechnology. Once a gene becomes mutated and dysfunctional in patients, providing a functional copy of that gene or gene products can help to improve the health conditions. Gene therapy endeavors to provide a functional copy of a gene in individuals who are carrying a defective gene. On the other hand, gene editing aims to correct a disease-causing gene, either by gene editing or altering the expression of the gene. Genetic counseling helps in the identification of disease-causing genes in the family history of a person and suggests measures to avoid the passing of the gene to the offspring. Additionally, innovative research in molecular biotechnology has immensely helped in the process of genetic counseling. The advent of whole genome sequencing and whole exome sequencing greatly helps in the process of genome profiling and identification of disease-causing genes in an individual. It also led the way for genomic medicine. In environmental science, use of living organisms to clean the environment is known as bioremediation. Genetically modified microorganisms, including bacteria and related products such as enzymes, are used to treat the environmental pollutants and/or hazardous substances present in the environment. Chemical substances emitted from various industries can act as environmental pollutants. As the turnover of these chemicals is very slow, these chemicals cannot be degraded fast by means of a natural chemical cycle. Biotechnological innovation led to the production of genetically modified organisms that can efficiently combat the environmental pollution due to industrial chemicals by degrading these into degradable products. A good example of it is the oil-eating bacteria, which can break down the hazardous chemical in the oil into small biodegradable molecules. Oil spills pollute marine water and the related ecosystem severely. Bioremediation using oil-eating bacteria has helped in improvisation of the pollution management due to oil spillage. Biofuel is another contribution of different varieties of seeds as well as related genetic information, is a good example of it. Another endeavor of molecular biotechnology includes the creation of extinct species using gene cloning technology and genome preservation of endangered and/or threatened animals. These are some of the great efforts to conserve the biodiversity of the earth using molecular biotechnology. Molecular biotechnology to make pollution less environment. Biological carbon fixation by using decomposition and fermentation of organisms, especially agricultural wastes, and plant products with high cellulose content, are the primary source of biofuel. Also, molecular biotechnology has helped in the process of biodiversity conservation. Seed bank which acts as a storehouse for collecting and preserving figure 12.7 production of biofuel using biotechnology and recycling of energy. The biofuel producing algae are grown in a fermenter and subsequently processed using biotechnological techniques for the production of biodiesel. As a bioproduct of this process, glycerin products are produced. Biodiesel causes less pollution as it is biodegradable and less toxic compared to petroleum diesel fuel. In forensic science, molecular biotechnology is used in forensic science for many purposes including DNA fingerprinting. Identification of criminals and DNA barcoding of animals are possible by using DNA fingerprinting technology. In short, during DNA barcoding, DNA is extracted from an organism followed by PCR amplification and sequencing of a particular DNA barcode region. Subsequently, the newly obtained DNA sequence is aligned to an existing reference database in order to find out a match in the sequence. DNA barcoding is helpful for taxonomic studies that identify new species of organisms including plants and animals. Figure 12.8 DNA barcoding helps in species identification. During DNA barcoding, genomic DNA is isolated from an organism. Using PCR technology, a specific region is amplified. After sequencing this barcode region, it is matched with the database to identify a new species. DNA barcoding is useful for taxonomic studies. Major breakthrough in molecular biotechnology. Humulin, diabetes mellitus is a deadly disease that causes lack of insulin. An effective treatment for diabetes is to supply insulin to the body externally. In the earlier days, insulin was produced from the pancreas of pigs and cows. However, at the beginning of the 1970s, the supply of animal-derived insulin was decreased, and as a result, the prevalence of insulin-dependent diabetes was increased significantly. To meet the crisis, Humulin came into the market. Humulin refers to the first recombinant human insulin that was produced in bacteria using rDNA technology. Since then, it has been highly successful to treat insulin-dependent diabetes and save the lives of millions of patients. For producing humulin, the human insulin-producing gene was genetically engineered and inserted into a vector. Subsequently, the host bacterium E. coli was transformed using the recombinant DNA containing the human insulin gene. Finally, large-scale cultivation of these genetically modified E. coli cells were carried out to produce and purify humulin by a pharmaceutical company in 1982. The production of humulin was a landmark discovery since it introduced a new era of pharmaceutical biotechnology. Figure 12.9 diagram showing the steps of human insulin production using rDNA technology. Human insulin-producing gene is isolated from the cells and cloned in a suitable plasmid using rDNA technology. The plasmid-containing vector is transformed into the bacterial cell, for example, E. coli, which starts producing insulin protein after being transformed. The human insulin-producing bacteria are harvested on a large scale in a fermentation tank followed by extraction and purification of human insulin. DNA fingerprinting. DNA fingerprinting refers to the unique identity of everyone's DNA. Each cell of our body is made up of DNA formed of the same basis, namely A, T, G, and C. However, there is a variation in the genomic DNA, which is unique to every individual. These variations in the DNA sequence are known as single nucleotide polymorphism (SNP). Therefore, if our genomic DNA is subjected to type 2 restriction endonuclease, it would cut the genomic DNA at different sites depending on the variation in SNP. It thus produces DNA fragments of different sizes, which is known as restriction fragment length polymorphism (RFLP). Surprisingly, it produces a unique pattern of RFLP for every individual. This forms the basic principle of DNA fingerprinting. Originally, in 1985, this technology was invented by Alex Jeffrey. There is another PCR-based approach that is used to detect the variation of flanking intronic sequences known as short tandem repeats (STR) or a variable number of tandem repeats (VNTR), and it produces a unique pattern in every individual's genome. Applications of DNA fingerprinting cover broad areas. It is useful to identify the criminals based on the biological samples, such as blood and hair found at the spot of the crime. Colin Pitchfork, a criminal was caught for the first time on the basis of pieces of evidence made by DNA fingerprinting technology. Additionally, it plays an important role in the determination of paternity and the identification of genetic disorders. Figure 12.11 steps of gene cloning, which enable creation of the sheep dolly. In brief, a somatic cell from a white-faced sheep was taken and it was fused with a nucleated oocyte from a black-faced sheep. The embryo from the fused cell was implanted in the body of a black-faced sheep that subsequently gave birth to a white-faced sheep. This indicates that the nuclear encoded information from the white-faced sheep was successfully cloned and transmitted to the newborn sheep. Later on, the reprogramming of cells using biotechnology was successfully carried out in other areas of medical research. For example, in 2007, scientists reprogrammed skin cells into pluripotent stem cells. These pluripotent stem cells can be differentiated into almost all the cell types found in humans. Thus, it offers huge potential for treating a number of diseases. CRISPR technology, a recent development in the field of biotechnology, is CRISPR-Cas9 technology for genome editing. CRISPRs. Figure 12.10 basic steps of DNA fingerprinting technology. After extracting the DNA from cells, restriction endonuclease enzymes are used to digest the genomic DNA into pieces of a specific size. When the digested DNA is run into an agarose gel, it produces a specific pattern of bands. The pattern of bands produces a unique identity for each individual. Identification of criminals is one of the applications of DNA fingerprinting technology. Dolly, gene cloning and reprogramming of cells. A domestic sheep was given birth in 1996 using cloning technology. Nucleus from somatic cells of the mammary gland was transferred into a nucleated ovum to clone Dolly, who was developed and reproduced normally. It was a remarkable event because it demonstrated that the creation of life is possible using somatic cell and gene cloning technologies. After this success, a number of other animals like horses, deer, and pigs have been cloned. In fact, recently extinct animals are also being produced with the help of cloning technology from the frozen tissue of that animal. For example, the Pyrenean ibex, a species of wild goat that was declared extinct, was cloned successfully in Spain. This offers numerous possibilities for maintaining the ecological balance. Clustered regularly interspaced short palindromic repeats are the short repetitive DNA sequences that are present throughout the genome. Cas9 CRISPR associated 9 is the enzyme that recognizes these repetitive sequences and acts as a molecular scissors to cut the DNA at a specific sequence recognized by the guide RNA GRNA. Similar to the restriction endonuclease system, CRISPR Cas9 is also used by bacteria for protecting themselves against the attack of viruses. Figure 12.12 the basic mode of functioning of CRISPR technology. Guide RNA contains a unique sequence that can target a portion of a gene. Cas9 is an enzyme that acts as a molecular scissor. When the guide RNA and Cas9 come together, it forms an active complex and cut a region of a gene. Once the guide RNA and Cas9 complex makes a cut, the gap in the DNA sequence is filled up with the help of the DNA repair machinery. The newly made DNA contains the desired sequence. CRISPR technology is widely used for the purpose of gene editing. The system has been adopted by the biotechnologists and successfully used for the purpose of genome editing. In brief, biotechnologists design and synthesize a guide RNA, which is complementary to a short region of the mutated gene of the patient. Then, along with the enzyme Cas9, the guide RNA is delivered to the cells of the patient. The guide RNA recognizes the mutated gene and Cas9 cuts the mutated DNA out of the genome. In brief, biotechnologists design and synthesize a guide RNA, which is complementary to a short region of the mutated gene of the patient. Then, along with the enzyme Cas9, the guide RNA is delivered to the cells of the patient. The guide RNA recognizes the mutated gene and Cas9 cuts the mutated DNA out of the genome. It is followed by healthy replacement of the mutated gene with a normal copy of that gene. In the recent past, human cells were subjected to the CRISPR-Cas9 mediated editing process for the first time. Subsequently, it has been demonstrated using animal models that CRISPR-Cas9 technology can be used for treating genetic disorders like cystic fibrosis. Figure 12.13 the promise of CRISPR technology in the treatment of genetic disorders. The diagram shows the basic steps of CRISPR-mediated gene editing using sickle cell anemia as an example. In brief, a genetic disease can be caused either by an inherited mutation or a de novo mutation in the disease-causing gene. Using a pre-designed guide RNA and Cas9 enzyme, gene editing is carried out. The guide RNA can identify the mutated DNA and makes the gene a target for the Cas9 enzyme, which cuts the mutated sequence out of the gene. Subsequently, it is followed by the replacement of a healthy copy of that gene. In this way, CRISPR-mediated gene editing technology can combat a number of genetic diseases. Technology. Bioengineers have successfully made a 3D printed model of human lung. Further research is going on to make the human heart, liver, and kidney as well as to overcome the other shortcomings associated with this technology. However, this tremendous success of bioengineers and biotechnologists can be a great boon for the patients who require organ transplantation with a high success rate. Figure 12.14 a model of a three-dimensional 3D bioprinter. Personalized medicine. Personalized medicine ensures maximum safety margin and the best possible treatment response while using a drug to treat a disease. Due to the advent of medical biotechnology, nowadays, advanced diagnosis, proper risk assessment, and optimum therapeutic management have been made possible. The application of genomic technologies immensely helps to make out the unique person-specific molecular genetic profile, which makes them prone to a particular disease. Similar technological advancement is used to augment the prediction algorithms for identifying the strategy of personalized treatments. For treating cancer and cardiovascular diseases, personalized medicine came up with great success. One of the most. Human Genome Project. Human Genome Project was an endeavor to sequence the whole genome of human being. It was initiated in the year of 1990 and was coordinated by the National Institute of Health and the Department of Energy, United States of America. In 2003, primary sequencing of the human genome was announced to be completed. Human Genome Project HGP for the first Human Genome Project HGP for the first time revealed that a human being contains approximately 2,500 genes. It has also been found that the over 3 billion base pair genome of a human contains 97.2% of non-coding DNA and the rest 2.8% of the genome codes for an mRNA and or protein. Out of this vast majority of non-coding DNA, 42.3% is the intronic region of the gene and the rest 54.9% constitutes intergenic regions. It was a major breakthrough as it revealed the blueprint of the genetic makeup required for the formation of human life. The Human Genome Project made a significant contribution to understanding the structure and function of human genes and their misregulation during the disease condition, especially in case of genetic disorders. 3D bioprinter. Another remarkable breakthrough that brings a high hope to millions of patients with organ dysfunctioning is 3D printed organs. In US alone, more than 1 million patients are waiting for an organ transplant. However, a conventional organ transplant has many drawbacks including a high possibility of organ rejection due to the body's immunity. Therefore, scientists have been thinking of reconstructing an organ from a patient's own cell in a laboratory setup. Due to the immune compatibility, the chance of organ rejection would be less in this case. The field of medical biotechnology and tissue engineering has come up with the exciting idea of 3D printed organs, where a bioprinter can be used to create organelles and eventually a complete organ. However, the most challenging job for the bioengineers, so far, was to create the network of vascularization that provides oxygen and food supply in the reconstituted organ. However, in the recent past, scientists have been successful to overcome this limitation. Using projection stereolithography, successful examples of personalized medicine usage was the treatment for breast cancer patients. Almost 30% of breast cancer cells express a protein called HER2. It makes the cancer cells non-responsive to standard drugs. A drug called trastuzumab was used along with chemotherapy and it showed a significant effect in treating the cancer cells. Figure 12.15 biotechnology lights up the path of personalized medicine and provides better management of complex disorders. Usually, a disease is treated with a medicine regardless of the genetic background of the patients. This results in a beneficial effect of the medicine for some patients but not for all. However, with personalized medicine, a drug can be chosen based on the disease as well as the genetic background of the person. This individualized therapeutic approach is expected to result in better management of the disease due to its precise specificity. Synthetic biology. One of the most recent breakthroughs of biotechnology is synthetic biology. The highly interdisciplinary branch of science aims to apply the principles of engineering for producing biological organisms. Specifically, synthetic biology is promising to design and produce living organisms using chemical and physical principles of life. After several trial and error processes, scientists have recently succeeded to produce a bacterial cell named Mycoplasma laboratorium. The 1.08 million base pair genome of the bacteria, Mycoplasma mycoides, was designed and synthesized by the scientists in vitro, followed by insertion of the genetic material into a mycoplasma cell with pre-destroyed genetic material. The newly transformed mycoplasma cell grew and divided normally and it was having the features of the newly synthesized genome. This was a remarkable event in the field of biotechnology because of its potential to produce bioactive molecules such as drugs and biosensors. Synthetic biology is a promising field due to its potential to produce synthetic genes, protein, and/or an organism that can be beneficial for human health and economical purpose. Figure 12.16 Synthetic biology, a groundbreaking contribution of biotechnology. Usually, a disease is treated with a medicine regardless of the genetic background of the patients. This results in a beneficial effect of the medicine for some patients but not for all. However, with personalized medicine, a drug can be chosen based on the disease as well as the genetic background of the person. This individualized therapeutic approach is expected to result in better management of the disease due to its precise specificity. Synthetic biology. One of the most recent breakthroughs of biotechnology is synthetic biology. The highly interdisciplinary branch of science aims to apply the principles of engineering for producing biological organisms. Specifically, synthetic biology is promising to design and produce living organisms using chemical and physical principles of life. After several trial and error processes, scientists have recently succeeded to produce a bacterial cell named Mycoplasma laboratorium. The 1.08 million base pair genome of the bacteria, Mycoplasma mycoides, was designed and synthesized by the scientists in vitro, followed by insertion of the genetic material into a mycoplasma cell with pre-destroyed genetic material. The newly transformed mycoplasma cell grew and divided normally and it was having the features of the newly synthesized genome. This was a remarkable event in the field of biotechnology because of its potential to produce bioactive molecules such as drugs and biosensors. Synthetic biology is a promising field due to its potential to produce synthetic genes, protein, and/or an organism that can be beneficial for human health and economical purpose. Figure 12.16 Synthetic biology, a groundbreaking contribution of biotechnology. A computer-designed genome is synthesized in vitro and assembled within a living cell, for example, in a single cell eukaryote-like yeast. Subsequently, the synthetic genome is used for making a fully functional synthetic cell. Synthetic cells can be used to produce synthetic genes and/or proteins for the benefit of humankind. Future of molecular biotechnology. Molecular biotechnology has been one of the most promising branches of biology. It has contributed significantly to human life and society. Being an interdisciplinary subject, biotechnology combines the knowledge of multiple disciplines for application in multiple fields such as agriculture, medicine, food, environmental issues, and many more. This led to the diversification of the field into broader areas such as agricultural biotechnology, pharmaceutical or medicinal biotechnology, food processing technology, and environmental biotechnology. In the 19th century, bioprocess technology, a branch of biotechnology, was used largely by the industries for the production of proteins, enzymes, and antibiotics. Fermentation technology was used to produce ethanol, butanol, and acetones using microorganisms for subsequent use in the downstream industrial processes. It greatly enhanced the demand and value of such technology for economic development as well. Nowadays, application-oriented molecular biotechnology has revolutionized the field by opening further avenues for numerous sectors such as agriculture, medicine, food processing, and so on. Based on rDNA technology, it offers the production of genetically modified organisms that would directly and/or indirectly benefit human life and society. In practice, rDNA technology together with bioprocess technology is used to produce numerous recombinant proteins and enzymes of therapeutic value by industries for subsequent human usage. Another most promising branch of biotechnology dealing with human health and related industries is hybridoma technology. Using genetic engineering, an antibody-producing B lymphocyte is fused with a melanoma cell. The hybrid cell is a cancer-free lymphocyte capable of producing large quantity of that specific antibody. This hybridoma technology is used for producing monoclonal antibodies (mAb) that are used as vaccines and for making diagnostic kits in the industry. Many of these antibodies, enzymes, and recombinant proteins are either in the process of a clinical trial or getting made and marketed by multiple industries worldwide. Medicine to combat a number of diseases such as cancer, arthritis, AIDS, heart disease, and neurological disorders are being developed using the tools of modern biotechnology. Diagnostic tools as well as products related to advanced medical divide and become confluent. Cells can be derived either by explant method or enzymatic method. In the explant method, a small tissue is placed in a glass or treated plastic plate and fed with appropriate culture media. After a few days, cells emerge from the tissue, grow, and become confluent. Next method is the enzymatic dissociation method, which is faster than the explant method. Here, the tissue is treated with proteolytic enzymes, trypsin, collagenase, disphase, etc., which help in the creation of a cell suspension. These cells are separated from tissue fragments and seated in a glass or plastic vessel containing culture medium and allowed to expand. Primary cells usually grow either as adherent or suspension culture, depending on their origin. Adherent cell culture. Cells are attached to the substrate or bottom and grow as a monolayer. Therefore, these are also known as anchorage-dependent cells. Once seated in a cell culture flask, these cells get attached to the bottom within a day, spread and take proper morphology. Upon reaching confluency, these cells need enzymatic or mechanical treatment for subculturing. Enzymatic treatment involves the use of enzymes like trypsin, whereas, during mechanical treatment, cells are detached from the bottom using a cell scraper. Adherent cells mainly include cells derived from various tissues, e.g., fibroblast cells, mesenchymal cells, and epithelial cells. Suspension cell culture. These cells do not adhere to the substrate and keep floating in the culture media. These are grown in untreated cell culture flasks and grow either in clumps or as single cell suspension. These do not need any enzymatic or mechanical treatment for subculturing and can be maintained easily without any frequent media change. However, these need agitation to maintain adequate gas exchange. It mainly includes lines derived from blood, lymphoblasts, hematopoietic stem cells, and tumor cells, etc. Applications have been designed, developed, and tested by the biotechnology companies. Use of genetically modified organisms including animal cells, plant cells, and microorganisms for producing substances for industrial usage is also a promising field. For example, productions of enzymes like protease, amylase, and lipase using biotechnological advancement are used in various industries including meat and leather industry, starch, as well as detergent industry. Genetically modified transgenic plants and animals are used for producing organisms with higher yields and good quality product. This is immensely helpful for agricultural purposes and subsequent supply of food. The technology of cut and paste to bring together two DNA pieces for making recombinant DNA with new properties have a great potential for both basic and applied research in biology. Although there are some ethical and social concerns regarding the usage of biotechnology, in general, its potential to augment the quality of human life is unquestionable. Additionally, biotechnology being a highly applied field offers the opportunity for numerous technological innovations, which can be immensely helpful for worldwide economic growth. Animal biotechnology. Basics of animal cell culture. Cell culture. Cell culture is defined as a process in which cells are grown and maintained under artificially controlled environment. In this process, cells are removed from the organ of an experimental animal either mechanically or enzymatically. Animal cell culture is divided into mainly primary cell culture and cell lines. Cell lines are further categorized into finite and infinite cell lines. Primary culture. Primary culture is defined as a cell culture in which cells are derived from the tissue of an organism, grown outside the body in vitro in a suitable environment where they grow. 2. Infinite continuous cell lines. When a finite cell line gets transformed, it acquires the potential to divide indefinitely and is known as an infinite cell line. These transformations occur either spontaneously or can be induced by chemical or viral methods. These are fast proliferating and can grow in multi-layers unlike finite cell lines and also in suspension culture. Continuous cell lines are usually aneuploid and require less serum for growth. This mainly includes tumorigenic cell lines like HeLa, HEP2, and Neopocket2. Cell strain. Cell strain is a lineage of cells originating from primary culture or cell line by positive selection or cloning or some other method. Subsequently, it may acquire additional genetic modifications from the initiation of its parent cell line. These exhibit euploidy and cannot proliferate indefinitely like continuous cell lines. All cells in a cell strain are homogeneous and exhibit the same phenotype, e.g., astrocytes, myocytes, keratinocytes, etc. Secondary subculture or cell line. When the cells of primary cell culture become confluent, they are subcultured by transferring to a new culture vessel, flask, petri plate, which contains fresh media and growth factors. These cultures are known as subculture or cell lines. Subculturing is done by detaching the cells using enzymatic treatment or manual scraping. Once the cells are in suspension, these are counted under a seated in a fresh culture vessel. These cell lines have been divided into the following two categories depending on the lifespan of the culture. 1. Finite cell lines. These cell lines have a limited lifespan and proliferate for a limited number of cycles before entering senescence. They get passage several times, slowly stop proliferating and reach senescence. These cells grow in monolayer and are anchorage dependent. These are usually diploid. The number of cells doubling time varies according to species, culture conditions including media composition, and oxygen level of the incubator, normoxia or hypoxia. Figure 12.17 Animal cell culture. Primary cell culture is done by either explant or enzymatic method. These cells either grow attached to the substrate of adherent culture or float freely in media suspension culture. As the cells become confluent, these are passaged using enzymes, trypsin EDTA, or manually using cell scrappers, lifters, and are known as secondary culture. Further passaging is known as tertiary culture and so on. Year historical events in the evolution of cell culture: 1885 maintenance of chick embryonic cells in saline culture. 1897 expansion of blood and connective tissue derived cells in serum and plasma. 1903 cell division of leukocytes derived from salamander in vitro. 1907 cultivation of nerve cells, isolated from frog, in a lymph clot held by the hanging drop, method and observation of the growth of nerve fibers in vitro for several weeks. 1910 long-term cultivation of chicken embryo cells in plasma clots. 1911 first liquid media consisting of seawater, serum. Embryo extract, salts and peptones were made. 1913 introduction of strict aseptic techniques so that cells could be cultured for long periods. 1916 introduced proteolytic enzyme trypsin for the subculture of adherent cells. 1923 development of Carl or T-flask is the first specifically designed cell culture vessel. 1927 production of the first viral vaccine Virginia. 1940 first time use of penicillin and streptomycin in cell culture medium to decrease. Microbial contamination. 1949 cultivation of poliovirus in human embryonic cells in culture. 1952 establishment of a continuous cell line from human cervical carcinoma, HeLa cells. 1955 establishment of the first chemically defined media. 1961 isolation of human fibroblasts, WI381. 1965 fusion of human and mouse cells using viral method. 1975 production of first hybridoma which can produce monoclonal antibody. 1978 development of serum free media using growth factors and hormones. 1982 first recombinant protein, human insulin got licensed as a therapeutic agent. 1986 lymphoblastoid gamma IFN licensed. 1987 tissue type plasminogen activator, TPA, from recombinant animal cells became commercially available. 1989 trial of recombinant erythropoietin. 1990 recombinant products in clinical trial, FGF, EGF, MAPS, and GM-CSF. 2007 reprogramming of human cells to iPSCs. Table 12.2. History of animal cell culture. CO2 incubators. CO2 incubators provide cell cultures with proper gases, pressure, humidity, and temperature for their growth and expansion. These are mostly water jacketed, equipped with gas cylinders, filters, thermostat, and a tray for water. It has shelves that can be removed for cleaning, and these incubators have an inbuilt mechanism for their sterilization. Microscopes. All the cell culture labs are equipped with basic inverted microscopes. These can be simple, bright field or large, fluorescent microscopes. These are used for routine observation of culture, for checking its growth or any sign of contamination, and for cell counting. Centrifuges. These can be very small spins, benchtop, or a large instrument. These are used for the isolation of cells and splitting of cell cultures, both suspension and adherent cultures. Most of these are refrigerated and provided with timers. Other instruments used in cell culture include pipettes, cell counters which can be manual or automated, autoclaves for sterilization of glassware and media, refrigerators for storage of media, growth factors, and reagents, pH meters, and liquid nitrogen containers for long-term storage of cell cultures. Culture conditions. Culture conditions may vary for every cell type, but the artificial environment for cell culture comprises of certain indispensable features. It mainly consists of 1. A substrate on which mostly adherent cells are grown, which may be coated in certain cases. However, some cells grow in floating condition as well, which are termed suspension cultures. 2. Cell culture medium. It provides nutrients for the growth of cells and has essential nutrients, amino acids, carbohydrates, vitamins, and minerals, hormones, and growth factors. 3. Physiochemical environment. It includes control of gases O2, CO2, N2, and pH, osmotic pressure, temperature, etc. Requirements of animal tissue culture lab. Laminar flow hoods. Laminar hoods provide a sterile area for cell culture practice. These are equipped with a glass cover which allows only the hands of the user to work inside it. The sterile air flows towards the user and prevents any flow of microbes or dust from room user towards the culture. These hoods are also equipped with UV tubes which are used to sterile the work area when not in use. Figure 12.18 Requirements of animal tissue culture. Sterile media and reagents. All the commercially available ready-made media and reagents undergo strict quality check methods which are mentioned by the manufacturers. These are sterile and can be used directly following sterile handling techniques. However, the reagents which are prepared in lab need to be autoclaved or sterile filtered before using them for culture. These are to be also checked for sterility before use. Personal hygiene and protective equipment. Hands should be washed thoroughly before and after working in cell culture room. Sterile lab coat, cap, shoe cover, mask and gloves should be worn to prevent contamination from skin, mouth and street clothes. Hair should be tied properly and cosmetics should not be worn. Advantages of animal cell culture. The most important advantages of animal cell culture are listed below. 1. Physiochemical pH, temperature, and dissolved gases and physiological conditions, nutrients and cellular interactions can be regulated and monitored easily. 2. It is easy to get homogeneous cell populations in cell culture in contrast to heterogeneous cell populations within a tissue. 3. Characterization of cells for cytological and immunological studies is easier in comparison to immunohistological studies of tissue samples. 4. These cells can be cryopreserved for long term and can be revived whenever needed. 5. It reduces the need for animal experiments. 6. As compared to in vivo experiments, a smaller quantity of reagents is required. 7. It is free of the ethical and moral issues associated with in vivo experiments. Limitations of animal cell culture. Animal cell culture is associated with a lot of limitations. Few of those have been listed here. A good tissue culture lab must possess a laminar flow hood, CO2 incubator, centrifuge, cell counter, pipettes, and microscope. All the glassware should be used after autoclaving and all the plasticware should be disposable and brought inside the lab after UV sterilization. Aseptic culture techniques. Animal cell culture is sensitive to many factors including purity of air, level of gases, pH of media, sterility of media, and sterility of work area. Therefore, to maintain good culture practice, aseptic techniques are to be followed. Aseptic techniques mainly cover the following areas. Sterile work area. Animal cell culture should be done in laminar flow safety cabinet. There should be a special room dedicated to cell culture and should have access to limited personnel. Ultraviolet light should be switched on before and after every user. Working area should be disinfected with 70% ethanol before and after every use. Inside the hood, only the reagents required for the specific experiment should be kept and it should be uncluttered. Waste should be discarded in bleach or any other disinfectant. Sterile handling techniques. Gloves should be worn before entering cell culture room and should be wiped with 70% ethanol before starting work. All the reagents and materials should be wiped with 70% ethanol before keeping it in laminar hood. Reagents and media should be kept in small volumes to avoid contamination. Reagents and media should be aliquoted using sterile pipettes and pouring should be avoided. Multiple flasks or bottles should not be opened at the same time. Two cell lines should not be handled at the same time to avoid cross-contamination. For other applications also because of ease of handling and economic nature as compared to animals, it can be used to directly observe the cytopathic effects of the virus and understand its infection cycle and mode of action as well. Cancer research. In cell culture, normal as well as cancer cells from all the tissues can be grown easily. Normal cells can also be transformed into cancerous cells by the use of radiation, viruses, and other chemicals. Thus, the effect of these factors can be utilized to understand the mechanism of cancer development. Further, cultured cancer cells also serve as a model system for determining suitable drugs and newer methods for selective destruction of various cancer cells. Toxicity testing and drug screening. Animal cell culture has been used extensively to test the effects of various drugs, molecules, and cosmetics on the survival and growth of a range of cell types of different origins. These cells are also used to identify the maximum permissible doses of potential new drugs before getting them tested in animal models. Genetic counseling. Fetal cells can be isolated from pregnant women using amniocentesis. These cells can be further cultured and used to determine any genetic defects. Karyotyping, chromosome banding, and other techniques can be used to determine fetal disorders at an early stage. Genetic engineering. Cultured cells can be introduced with foreign DNA or RNA to study the effect of new proteins on cells. Insect cells have widely been used as mini cell factories for the expression of substantial quantities of proteins after infecting them with genetically engineered baculoviruses. Gene therapy. Gene therapy is a technique in modern biology which involves insertion of a gene clone altered into cells using recombinant DNA. 1. Need for extensive expertise and technical skills for regular practice of tissue culture. 2. Establishment of a cell culture facility, equipment, and other facilities are very expensive. 3. The cost of production of cells is very high than the direct use of animal tissues. 4. Difficulty in control of the external environmental factors temperature, pH, CO2, O2, and disposal of biohazards is difficult. 5. In cell cultures, cells are maintained in two-dimensional cultures. However, in vivo, they exist in three-dimensional organizations. Due to this, their cellular contact interactive features are lost. 6. Every tissue is composed of many cell types. However, cell lines can represent only a few cell types not mimicking the exact in vivo conditions. 7. Over a period, the genetic makeup of continuous cell lines may get altered, leading to the heterogeneity of cells. Applications of cell culture technology. Model systems. Animal cell culture acts as a reliable model system for understanding 1. Basic cell biology, biochemistry, and immunology. 2. The interactions between cells and pathogens. 3. The effects of various drugs on cells. 4. The beginning, process, and stimulus for aging. 5. Nutritional studies. Vaccine production. Vaccine production is one of the most studied areas utilizing animal cell culture research. The capacity to grow large number of viruses in cell culture has been used for the development of the polio vaccine followed by vaccines for many other diseases, like rabies, hepatitis B, chickenpox, and measles. Virus cultivation and study. Apart from vaccine production, cell culture is widely used for the propagation of viruses in patients with severe burns, deep skin injuries, and ulcers. Several patients with burns have been treated using lab-generated products. Research is also going on making artificial organs like the pancreas, kidney, and heart. Discovery of induced pluripotent stem cells (iPSCs) has enhanced the work in the organ culture development field. In research, both adult and embryonic stem cells are used. Embryonic stem cells, however, have ethical issues and thus are used for mainly understanding the pathways underlying differentiation into different lineages. Whereas, adult stem cells are devoid of such issues. Therefore, for patient care, adult stem cells are being explored widely. These cells have the potential to differentiate into a variety of different types of cells and organs. These are believed to be helpful in treating a number of medical conditions upon proper control of development and differentiation. And differentiated adult stem cells have already been used in many diseases like myocardial infarction, Parkinson's disease, diabetic foot ulcer, and diabetes. Technology. It can be used to replace defective genes, non-functional genes, or to deactivate improperly functioning genes. Gene therapy is mainly of two types: germline gene therapy. Defective gene and germline cell in the modified gene is passed to next generation, and somatic gene therapy. Defective genes repaired or replaced in some or all body cells of an individual, but the modification is not passed to the next generation. Genetically engineered protein. Animal cell cultures are being used to generate a number of proteins at a commercial level, which are essential to good health. However, some people cannot produce these because of genetic defects. It includes monoclonal antibodies, insulin, growth hormones, blood clotting factors, etc. Replacement tissue or organ. Animal cell culture can be used to replace tissue or organs. For example, artificial skin is being made in labs which can be used to treat. Figure 12.19 Applications of animal cell culture technology. Some model organisms are similar size to humans. Some organisms have more than 90% similar genes to humans. Model organisms can be used for the creation of detailed genetic maps. An ideal model organism should be relatively simple organisms, easy to maintain, and breed in laboratory settings, inexpensive, and easy to care for. The most common model organisms being used at present is for the Genetic Society of America include rat, Rattus mouse, Muse musculus, yeast, Saccharomyces cerevisiae, fruit fly, Drosophila melanogaster, nematode worm, Caenorhabditis elegans, Arabidopsis, chicken, Gallus, zebrafish, Danio, zebrafish, xenopus, fish and yeast, Schizosaccharomyces, pomba, fungus, nurse, crassa, water fleas, Daphnia, Dictyostelium, Discoideum. It is mainly used for vaccine production, drug testing, virus production, and production of genetically engineered proteins. It is also being used in gene therapy and the design of cell and scaffold-based tissues and organs for replacement in near future. Model organisms in biomedical science. A model organism is defined as a non-human species which can be extensively explored to understand various cellular, molecular processes, and mechanisms in humans without causing any harm to human beings. According to NIH, a model organism is an animal, plant, or microbe that can be used to study certain biological processes. Research development done in model organisms can be extended to humans because of the presence of similar genes which function in the same manner in both model organisms and humans. Need for model organisms in genetic research. These model organisms can breed in large numbers and also some possess a very short generation time. Figure 12.20 Model organisms in biomedical science. Bacterial restriction enzymes which cut the DNA at sites with specific nucleotide sequences. Restriction enzymes cut the DNA at unrelated locations, leading to fragments of different lengths. This makes the basis for the analysis of RFLPs. Restriction polymorphism occurs when an existing restriction site is removed or a new restriction site is created by some mutation. These alterations can be identified using a hybridization probe. RFLPs are identified by separating the DNA fragments of different sizes using gel electrophoresis and further its transfer to a nylon membrane, southern blot. The polymorphisms are then visualized as hybridization bands, which are different in the individuals carrying different allelic variants for a locus. It has various advantages including the production of codominant, also known as semidominant markers, which help to discriminate homozygotic and heterozygotic states in diploid organisms, stable and reproducible and selective neutrality. Its disadvantages include long methodology, labor-intensive, the requirement of high quality, and large quantities of DNA. RFLPs have also limited the identification of the whole genome variation in animals and reduced variability observed in domestic animals by inbreeding makes many RFLP sites non-informative. Oligonucleotide fingerprinting. In this method, hybridization of probe, e.g., genomic or synthetic oligonucleotide, is carried out for the different families of hypervariable repetitive DNA sequences, namely minisatellite, simple repeats, variable number of tandem repeats, VNTR, and microsatellite to reveal highly polymorphic DNA fingerprinting patterns, DFP. DNA fingerprinting was invented in 1985 by Sir Alec Jeffreys. Rather than simply identifying a species or some particular trait, it is particularly utilized for the identification of a specific individual. At present, it is being used both for the identification of paternity or maternity and for the identification of criminals or victims. A DNA fingerprint is prepared by digesting its DNA with a restriction enzyme, followed by electrophoresis in southern. It includes all the non-human species which are being used to study cellular and molecular processes occurring in human body. These include prokaryotes, invertebrates, and vertebrates like bacteria, fungi, fruit fly, mosquito, fish, rat, mice, rabbit, and dog. Molecular markers in livestock. A molecular marker is defined as a site where differences in the DNA sequences are found among members of the same species, which are associated with certain characteristics. It mainly represents polymorphism at DNA level. These differences may include insertions, deletions, translocations, duplications, and point mutations. These have characteristic biological properties that can be detected and analyzed in any body part, including blood or tissue at any stage, and these are also not confounded by the environment, pleiotropic, or epistatic effects. An ideal molecular marker should be highly polymorphic, codominant, frequently occurring in the genome, selective neutral behavior, easily available, easy and fast assay, and highly reproducible. Types of molecular markers in livestock. Molecular markers have been categorized into the following three types depending upon the type of method being used. Hybridization-based DNA markers. Hybridization-based DNA markers use cDNA, clone DNA elements, or synthetic oligonucleotides as probes. These probes are then labeled with a radioisotope or with conjugated enzymes that catalyze a color reaction to hybridize DNA. This hybridized DNA is then either cleaved with restriction enzymes or amplified by PCR. These are mainly of two types. 1. Restriction fragment length polymorphisms, RFLPs. 2. Oligonucleotide fingerprinting. Restriction fragment length polymorphisms, RFLPs. RFLP was developed in 1980 for the identification of the differences in the structure of DNA using requires very less samples. However, it detects polymorphism only as the presence or absence of a band of a certain molecular weight, and reproducibility of data may be inconsistent, and some primers may also show unspecific non-reproducible binding. Simple sequence repeats are microsatellites, SSRs. Simple sequence repeat SSR loci or microsatellites are also known as variable number of tandem repeats, VNTRs, and simple sequence length polymorphisms, SSLPs. These are present throughout the genomes of most of the eukaryotes and also in prokaryotes to a lesser extent. The length of SSRs varies from 1 to 6 nucleotides in length, and depending upon that these are designated as mono, D, tri, tetra, penta, and hexanucleotide repeats. They are repeated usually 5 to 20 times in the genome with a minimum repeat length of 12 base pairs. The tandem repeated sequences have been found to occupy large distribution of the size of the repeat unit and the length of the repeated locus. The microsatellites and minisatellites together constitute the variable number of tandem repeats, VNTRs. The lengths of particular microsatellite sequences tend to be highly variable among individuals, which make up molecular alleles. Every microsatellite has unique sequences which are used to make specific PCR primers. DNA is isolated from organisms, and many different microsatellites with specific primers can be amplified. The amplified DNA can be run on a gel and labeled to generate a unique genetic fingerprint. Microsatellites have emerged as one of the most studied and popular markers in livestock genetic characterization studies during the last few years. These are mainly used in animal stocks for evaluation of their genetic resources, parentage determination, disease research, determination of genetic variation within and among breeds, population substructure determination, reconstruction of phylogenetic relationships among populations, and further historical studies of domestication and migration of breeds. These markers are hybridization with a probe specific for a highly variable region in order to generate a large amount of polymorphism. In the case of human beings, many satellite DNAs are used as probes. Its main advantage is that the results cannot be altered. However, it requires standardization to be universally accepted as a reliable tool. PCR-based DNA markers. This technique is easier than probe-based methods and has led to the discovery of several useful and easy-to-screen methods. These markers have been further subcategorized depending upon the type of primers which is being used for PCR. 1. Random amplified link polymorphic DNAs, RAPDs. 2. Simple sequence repeats or microsatellites, SSRs. 3. Amplified fragment link polymorphisms, AFLPs. Random amplified link polymorphic DNAs, RAPDs. During the last decade, the RAPD has emerged as one of the most commonly used molecular techniques to develop DNA markers. It is a fast and efficient screening method for DNA sequence-based polymorphism at a very large number of loci. It works on the principle of using a single, short, arbitrary, and oligonucleotide primer, which binds to many different loci and produces relatively detailed and complex DNA profiles for detecting AFLP between organisms. It majorly uses one short oligonucleotide comprising of 8 to 10 nucleotides. It uses multiple primers with the most efficient and successful ranging from 5 to 21 nucleotides. The amplified products obtained range from less than 10 to over 100, depending on the primer-template combination and ratios. In this manner, a unique spectrum of products characteristic for each template and primer combination is developed, which is easily resolved and visualized using polyacrylamide gel electrophoresis and silver staining. It is simple and quick, cost-effective, produces a large number of bands, independent of any prior DNA sequence information, and markers are dominant, and the presence of a band could mean that the individual is either homozygous or heterozygous for the sequence. DNA chip and sequence-based DNA markers, single nucleotide polymorphisms, SNPs. It is defined as sequence polymorphism occurring due to the presence of a single nucleotide mutation at a specific locus in the DNA sequence. This mainly includes single base transitions, insertions, and deletions, and transversions. Among these, transitions are the most common. Its mechanism mainly includes the hybridization of detected DNA fragments with high-density DNA probe arrays, also called SNP chips. These markers are present in coding, and as well as non-coding regions of the genome and are present at 1 SNP in every 1000 BP. They are p-allelic markers, indicating a specific polymorphism in 2 alleles only of a population. Coding region SNPs can be directly associated with the protein function and possess stable inheritance pattern, making them more suitable markers for selection over time. It also possesses a very low mutation rate, is high stability, and detects the level of variation within a species. Further, it follows patterns of evolution, can differentiate alleles of disease, genes, and help in the creation of designer pharmaceuticals. But at the same time, the information obtained is less than that of a highly polymorphic microsatellite, which can be compensated using a higher number of markers, SNP chips, and whole genome sequencing. Marine biotechnology. Marine biotechnology is a field where products are derived or developed from a wide variety of marine animals and plants. This area also includes aquaculture, where aquatic organisms are grown in culture and used for food, fuel, cosmetics, pharmaceuticals, and other products. Marine biotechnology has extended its benefits in areas including pharmaceuticals, novel industrial enzymes, food production, biosensors, biomolecules, drugs, and new chemical compounds. Highly abundant in the genome and possess an extremely high degree of polymorphism and can be detected easily. Its advantages include very low template DNA, 10-100 nanograms requirement. These are distributed randomly throughout the genome, represent codominant markers, allele sizes can be determined with high accuracy, and the results are highly reproducible. But it has very high initial development costs, time-consuming and expensive, does not give any information about functional traits, biodiversity. Heterozygotes may be misclassified as homozygotes when no alleles occur due to mutation in the primary allele sites, and genetic divergence may be underestimated due to homoplasy using different forward and backward mutations. Amplified fragment link polymorphisms, AFLPs. AFLP was developed by Kegen in the early 1990s. It is a PCR-based method where genomic DNA is digested with restriction enzymes, followed by ligation of obtained restriction fragments with adapters at their ends. PCR primers are synthesized by making use of adapter sequence, the restriction site sequence, and a few nucleotides inside the restriction site fragments. These fragments are run on agarose gel electrophoresis, either through autoradiography, fluorescence methodology, or automated capillary sequencing instruments. AFLPs are dominant p-allelic markers. Some of these fragments are selectively amplified with 2 PCR primers having corresponding adapter and restriction site-specific sequences. Variations of many loci can be arranged simultaneously to detect single nucleotide variations of unknown genomic regions, in which a given mutation may be frequently present in undetermined functional genes. The AFLP method is an ideal molecular approach for studying population genetics and genome typing. It is also being widely utilized for the detection of genetic polymorphisms, evaluation, and characterization of animal genetic resources. Further, it is a cost-efficient, fast, reproducible, relatively inexpensive, and highly variable methodology. However, these Eucuma and Capopicus, alginate, and agar. Agar is extracted commonly from red algae like Gillium and Gracelaria. It is made up of mainly two important constituents, namely agarose and agaropectin, which form the backbone of the cell wall of certain algae. It is used as desserts, in preparation of microbiology culture media, packaging, leather industries, preservation of foodstuffs, paper manufacturing, laxative, dairy, and cosmetics industries. Carrageenans. Carrageenans are majorly derived from red and purple seaweeds and consist of a mixture of polysaccharides. These are used in the food industry based on their emulsifying and thickening properties. These can be found in diet foods including salad dressings, starch-free desserts, jellies, jams, syrups, and pudding sauces. These are also used as stabilizers of emulsions by the cosmetic and pharmaceutical industries. Alginates. Alginates are polysaccharides present in the cell wall of brown algae. These are rich in salts of sodium, calcium, or potassium alginate. Alginic acid is commonly extracted from laminaria, aconia, and macrocystis. Alginates are used for many biomedical applications in wound healing, transplantation of cells, and as emulsifiers and emulsion stabilizers in creams and lotions. Mollusca-derived products, biomolecules. Mollusca are known to be one of the most diverse groups of animals present on Earth. These are a group of organisms that possess soft bodies with a defined head and a foot region and possess a hard exoskeleton. These have been utilized for ages as a source of food, jewelry, tools, and even pets. Elastatins are obtained from the Indian Ocean sea hare. These are meiotic inhibitors, used as anti-cancer drugs in case of breast and liver cancers, solid tumors, and some leukemia. Spitulocine. About 71% of the Earth's surface is covered by oceans. The organisms inhabiting oceans are found in the greatest extreme weather conditions of temperature, light, and pressure. These marine organisms contribute to several processes having direct and indirect effect on human life. The existence of these organisms in varied and extreme environments contributes to the rich biological and genetic diversity. This variety has been found beneficial in various biotechnological applications including drug discovery, environmental remediation, increasing seafood supply, safety, and providing new resources for industrial processes. In recent years, biotechnologists have shown immense interest in marine life because they represent a major share of the Earth's biological resources and also because marine organisms have unique genomes, physical structures, metabolic pathways, reproduction organs, sensory, and defense mechanisms due to their ability to survive in extreme environments. Because of survival in this wide range of environments, they have acquired unique properties and bioactive compounds that are unparalleled by their terrestrial counterparts in some cases. Seaweeds products in food industry. Seaweeds are marine macroalgae or plant-like organisms generally found attached to a solid substrate in the marine environment. These lack true stems, roots, and leaves, but can perform photosynthesis using sunlight. Most seaweeds are red, e.g., dulse, and Nordic brown kelp, and combo or green, e.g., sea lettuce and sea grapes. Red and brown seaweeds are mostly marine, whereas green algae are also found in fresh waters. Seaweeds are an abundant source of food, fodder, and a reservoir of important chemical compounds useful in industry. Red algae and brown are the most valued seaweed, which hold a major share of human food throughout the world. These are rich in polysaccharides which help to maintain gut balance. The other edible seaweeds include Gracelaria, Undaria, Laminaria, and Caulopa. Industrially important seaweeds for carrageenans include species like chondrus, acids, hormones, and iodine is used for this. It acts by increasing the metabolism rate and stimulating oxygen consumption in the cells along with alleviation of heat production. Other useful compounds obtained from seaweeds include terpenes, amino acids, phenols, pyrulic substances, arsenic sugars, sterols, colorants, and amino acids. Spirulina, the blue-green bacteria, and Ascophyllum nodosum are used as diet supplements, general tonics, and rejuvenators. Some extracts have also been shown to possess antiviral properties. For e.g., carrageenans can inhibit herpes simplex virus, HSV, and human immunodeficiency virus, HIV. Many other seaweeds and their products have been shown to directly benefit human health. For instance, laminaria provides iodine, Sargassum nudicum provides vitamin E, and vitamin K, lithiamine, and phenolphthalein are rich sources of calcium carbonate and trace elements. The compound, monolite, isolated from specific sponge, has given a number of chemical analogs, many of which are found as potent as anti-inflammatory agents. Enzymes. Several enzymes have been isolated and characterized from marine bacteria. These bacteria are found to thrive in extreme temperature, pressure, salt, and lighting conditions, which in turn provides them with unique features enabling them to thrive in extreme environments. Depending upon their survival, these enzymes are found to be resistant to extreme heat, temperature, and salt. The most important thermostable enzymes include polymerases, ligases, gyrases, and restriction endonucleases. For instance, enzyme Taq polymerase was isolated from a heat-resistant bacterium, Thermus aquaticus. This thermostable enzyme later became the backbone of polymerase chain reaction. Other enzymes include collagenase, proteases, holoperoxidase, agalactosidase, etc. Biodegradable polymers. Microbes produce polyhydroxyalkanoates, PHAs, in the presence of rich carbon sources. Is a novel anti-tumor compound derived from Arctic clam fistula. Phase 1 clinical trials have been started to test its potency. Cyconitide is a novel non-opioid drug, which is isolated from the venom of fish-eating cone snails. It is being used in pain management and its effect is being tested in vitro and in vivo. Depsipeptide compound is found in small quantities in the mollusc Elysia rufescens and in the alga on which it feeds. This compound has shown anti-cancer properties in case of tumors of the lung and colon. Bryozoan-derived therapeutics. Bryozoans are a phylum of aquatic invertebrate animals. These are found in colonies and the skeleton of colonies is made up of calcium carbonate. The bryozoan, Bugula neridina, has been found to be a potential source of drug against leukemia, which is present either in or on the animal. Bryozoans are found to exist in a symbiotic relationship with a bacterium, where the bacterium synthesizes its drug as toxin to protect bryozoan from, and in return gets space to grow. Bryostatins include macrolide lactones isolated from marine bryozoans Bugula neridina. These are potent protein kinase modulators. They have been found to have roles as anti-cancer agents, anti-AIDS, HIV agents, and in Alzheimer's disease. Products from marine vertebrates. A spiny dogfish, Squalus acanthias, has been found to produce a small aminostraw molecule from its stomach and liver. Initially, it was reported to have broad-spectrum antibiotic activity. It has also been found to act against aberrant neovascularization by inhibiting angiogenic growth factors, including vascular endothelial growth factor, VEGF, platelet growth factor, PDGF, and basic fibroblast growth factor, BFGF. Therapeutic agents. Seaweed extracts have widely been used as therapeutic agents to detoxify human body cells and maintain skin pH. Laminaria digitate, which is a rich source of vitamins A, E, C, B, amino-specific fish with desired traits and then reproduction. This process is very slow and unpredictable and may not meet the increasing demand. On the other hand, transgenic involves the selection of desired traits, construction of their genes and incorporation in a selected brood to get progeny with those traits. Transgenic fish production is much easier and economical as compared to raising transgenic large animals or mammals. The reason for this is the small life cycle of fish and the laying of thousands of eggs, which helps in the generation of a large number of fishes with uniform genetic material. For example, zebrafish produces 1, 50, 400 eggs, and Atlantic salmon produces 50, 15,000 eggs. Disease resistance. Molecular biology has been instrumental in providing us with information about life cycles, disease transmission, its effects, host immunity, susceptibility to diseases, and antibiotic resistance. This understanding of molecular biology holds immense importance in aquatic industry. For instance, fishes are cultured in high density to meet seafood production, which puts a lot of stress upon fishes, increasing their vulnerability to a variety of infections. In such cases, any outbreak may lead to a heavy toll on the whole process, leading to huge losses to the industry. However, the development of resistant strains to such conditions may withstand a variety of diseases. Several transgenic approaches, including antisense and ribozyme technologies, have been introduced to promote disease resistance capacity in fishes. For instance, neutralizing the hematopoietic necrosis virus (HNV), which is responsible for mortality in salmonids, can drastically improve salmoniid growth. Other methods of expressing viral coat proteins in the host membrane have also been beneficial. However, the most effective method is to boost the immune system of the host organism by the expression of antimicrobial and antibacterial substances, which mainly includes antibacterial peptides like magnans and lysozyme, and essential minerals like phosphorus. Under aerobic conditions, these PHAs can be completely degraded up to 100% into water and carbon dioxide into methane in anaerobic conditions by microorganisms' existence in soil, water, and sewage. A special type of biopolymer lipid-like inclusion exists in the cytoplasm of bacterial PHAs. At present, there exist more than 300 species of gram-positive and gram-negative bacteria that act as a reserve energy material under phosphorus or nitrogen-limiting concentration with an excess of carbon sources. These species include Azotobacter crucatum, Bacillus megaterium, etc. Marine resources hold a large treasure which remains untapped. It holds immense potential for exploration and utilization by humankind. Its proper understanding and harvesting can lead to boom in areas like pharmaceuticals, bioremediation, biofilms, seafood, and biomaterials. Aquatic biotechnology. Aquaculture. Aquaculture has been defined as the culture of aquatic organisms including fish, mollusks, crustaceans, and aquatic plants by Food and Agriculture Organization (FAO). Or it can be defined as the manipulation and improvement of production of aquatic beings for supporting seafood industry. Use of biotechnology tools has emerged as an important tool to enhance the production of aquatic animals and comes under aquatic biotechnology. Increasing seafood demand and a gradual decrease in seafood harvest from fisheries has led to boom in aquatic biotechnology. The model tools of biotechnology help in the improvement of health, reproduction, development, and growth of aquatic organisms, and thus promoting the interdisciplinary development of environmentally sensitive and sustainable systems. Transgenic fish. Conventional fish breeding is being used for many decades and involves the selection of technologies, DNA fingerprinting, improving the precision and efficiency of transgenic techniques, and cryopreservation of gametes and embryos. Animal cytogenetics. Cytogenetics is defined as the study of genetic phenomena of an organism through the cytological analysis of chromosomes under light or electron microscope. Since 1964, after the discovery of Robertsonian translocation involving chromosome 1 and 29 of cattle karyotype, the application of cytogenetics in domestic animals has been expanded and utilized in every aspect. At present, our knowledge of chromosomes has been enhanced by improved chromosome banding techniques and it has been extensively utilized to establish similarities or differences among different species. Further, with the advancement of techniques and synthesis of DNA probes and the establishment of fluorescence in situ hybridization fish technique, our knowledge of human as well as domestic animals' chromosome structure and function has also increased. It is difficult to visualize the detailed structure of chromosomes under light microscope. Therefore, special staining techniques have been developed that bind with DNA and help cytologists study every detail and pattern of chromosomes efficiently and effectively. Based on the type of DNA stain used and pattern obtained, the available banding techniques which are used to study chromosome patterns and abnormalities have been categorized mainly as Q banding. This is the first banding technique described in 1970 by Torbjorn Kaspersen and his colleagues. In this method, chromosomes are stained with quinaprine dye, which works by alkylating the DNA. This dye produces characteristic and reproducible banding patterns for individual chromosomes. However, its fluorescence fades very quickly. G banding. It is also known as Gienza banding, wherein chromosomes are treated with trypsin and then stained with Gienza stain. Heterochromatin structures which are rich, stain darker than GC-rich regions, being freeze-resistant fish. Antifreeze protein AFP can be transferred to fishes to confer freeze resistance using recombinant biology techniques. These AFPs can be found in cold water marine fishes like winter flounder and ocean trout, which help in the prevention of ice crystal formation in the blood and thus protects from freezing. This gene can be transferred to commercial fishes like Atlantic salmon, which otherwise cannot survive sub-zero temperatures. This can be very beneficial for the fish industry. Growth rate. The growth rate in fishes can be enhanced either by giving them microinjection of growth hormone genes into fertilized eggs or by inserting early stage fish embryos with an extra copy of the growth hormone gene. Reproduction. Reproduction has emerged as a major issue in the fish aquaculture industry. The growth rate of fishes decreases with maturity along with the quality of the flesh. Using biotechnology tools, the maturation process can be delayed to keep up the quality of such fish. Along with this, non-reproductive sterile species can also be produced by modifying their genome. Such species do not possess any risk of farm-to-wild interaction and can also help in the reconstruction of stock of conserved sperm and stock identification by providing gene markers. Retroviruses have been constructed which help in transfer of gene of interest in marine organisms. Conservation. Molecular tools can be used to analyze the genome of many endangered species. They have also helped us in deciphering the molecular basis of gene regulation, expression, inhibition, and sex determination. These tools can further help in the upgradation of methodologies being used for defining species, stocks, and populations. The molecular approaches to achieve this include developing marker-assisted selection to do karyotyping of non-dividing cells. Further, it helps to determine how many copies of a specific segment of DNA are present in a cell. CH chromosomal microarray analysis (CMA) is the latest technique having resolution and efficiency to detect chromosomal imbalance, higher than existing banding techniques for fish. Fluorescent labeled DNA of test and control samples are arranged in a particular manner on a glass slide which are read by a special scanner. It can detect a genetic condition which are missed by conventional methods and has 100-fold higher resolution than karyotyping. It can identify tiny, submicroscopic deletions or duplications of DNA sequences, known as copy number variants, small chromosomal rearrangements, small duplications of chromosomal material, or small deletion of chromosomal material. Microbes in animal health improvement. The world population is estimated to reach 9 billion by 2050. The demand for food of plant and animal origin is constantly growing with the increasing human population. In past, to increase the production of high-quality meat, eggs, fish, etc., antibiotics and other medicinal products have been widely used. However, their long-term use has led to the development of antibiotic-resistant microbes which are harmful to end users' health as well as the environment. This increasing awareness has led to minimizing the use of antibiotics in animal feed and introducing the use of prebiotics and probiotics in animal feed. The viable option is to investigate and use sustainable additives to affect animal performance and welfare favorably, particularly through the modulation of the gut microbiome, which plays a critical role in the nutrition and health of host. This is because a balanced gut microbiota constitutes an efficient barrier against pathogen colonization, produces useful metabolites, for example, proteins, vitamins, and short-chain fatty acids or SCFAs, and stimulates the immune system in a non-inflammatory manner. In this context, the use of probiotics, prebiotics, and symbiotics could be a possible alternative. These additives are aimed at increasing resistance against transcriptionally less active. This technique produces reproducible patterns every time and is being used worldwide. G banding was first time reported for species including pig, cattle, horse, goat, rabbit, and cat. R banding. This is also known as reverse banding, where staining is done using genes only. In this method, chromosomes are heated before staining, which preferentially melts the rich region which stains strongly during G banding. This allows GC-rich region to take up stain and produce patterns that are reverse of G banding. This is mostly used to provide critical details about gene-rich regions that are located near the telomeres. C banding. C banding is used to stain mainly constitutive heterochromatin and centromere. It is a special beam sustain. T banding. T banding is used to stain mainly telomeres. In this method, chromosomes are stained with either phosphate buffer or Rose-balanced salt solution before staining with beam sustain. CT banding. CT banding is used to stain both heterochromatin and centromere and telomeres. Here, chromosomes are first treated with barium hydroxide before proceeding for beam sustain. Molecular cytogenetics. Molecular cytogenetics includes the use of both molecular biology tools and cytogenetics to differentiate between normal and abnormal genotypes. Two techniques used in molecular cytogenetics include fluorescence in situ hybridization FISH and comparative genomic hybridization CGH. Fluorescence in situ hybridization FISH. It refers to the use of fluorescently labeled probes to hybridize with cytogenetic cell preparations. It was developed in 1986 by Kinkle et al. It is used mainly to determine the number of copies of a specific gene segment in a cell. The fluorescent tags used in FISH are safe and very simple to use. Also, these can be stored for indefinite periods. The probes are available for humans and mice, but for cattle these have to be synthesized. FISH can detect chromosomal abnormalities such as submicroscopic deletions and aneuploidies which are not feasible with existing banding techniques. Also, it can help feed intake and digestion. Probiotics increase activity of intestinal enzymes, produce enzymes like amylase, lipase, cellulase, and protease which help proper digestibility of nutrients. It also increases the height of intestinal villi, thus increasing the total surface area available for absorption of nutrients. 4. Role in disease and rotavirus control. Lactobacillus secretes vitamins B, some essential amino acids and enzymes that help in digestion. It also produces high quantity of lactic acid which can inhibit Helicobacter pylori, preventing gastritis, peptic ulcers, and gastric cancer. It also helps in the treatment and prevention of infectious diarrhea caused by rotavirus. 5. It also aids in weight gain. Prebiotics. The concept of prebiotics was introduced in 1952. It is defined as non-digestible food ingredients that beneficially affect the host by selectively stimulating the growth and/or activity of one or limited number of bacteria in the colon, or a substrate that is selectively utilized by host microorganisms conferring a health benefit. A dietary substrate should fulfill the following criteria to be classified as prebiotic. 1. It should be resistant to digestion in the upper alimentary canal. 2. It should be fermented by microbes present in the intestine. 3. It should be stable during various food processing procedures. 4. It should lead to selective stimulation of the growth of probiotics. The prebiotics are nutrient supplements for some specific gut microbes. Mostly, these are composed of carbohydrates, proteins, peptides, and lipids. The natural source of prebiotics is cereals, fruits, and legumes. However, few formulations have been also chemically synthesized. Most commonly used. Colonization of pathogenic bacteria, enhancing host immunity, improving the health status of animals, and minimizing the load of foodborne pathogens. Probiotics. According to the Food and Agriculture Organization (FAO) of the United Nations World Health Organization, probiotics are defined as live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. Probiotics have shown beneficial effects on health and stimulation of growth which makes them broadly used in animal feed. The probiotics mainly include bacteria, yeast, and molds. Bacteria. Lactobacillus, acidophilus, sporogenes, plantarum, rhamnosum, delbruch, rotary, fermentum, lactis, celebiosis, brevis, casei, parsimonus, paracasei, gasseri, and crispattis. Bifidobacterium, bifidum, infantis, adolescentis, longum, dermaphyllum, breve, lactis, and animalis. Streptococcus, lactis, chromorus, salivarius, intermedius, thermophilus, diacetylactis leuconostoc mesodroids, pediococcus propionibacterium, bacillus enterococcus, and enterococcus faecium. Yeast and molds, Saccharomyces cerevisiae, Saccharomyces boulardii, Aspergillus niger, Aspergillus oryzae, and Candida pintalopsi. Probiotics have a number of important roles, few of which have been summarized below. 1. Competing with pathogenic bacteria. Probiotics change gut flora by outcompeting undesirable microbes and antagonism. They stick to the intestinal wall preventing pathogens to enter the bloodstream. 2. Lactose intolerance. It occurs because of the enzyme cysteolactosidase which degrades the lactose into monosaccharides. So, when milk products are produced using bacteria S. thermophilus and L. delbrueckii sp. vulgaris having high galactosidase activity, this problem can be solved. Out of all these, most promising is non-digestible oligosaccharides (NDOs). These prebiotics when used in excess can be harmful, and to make maximum use of it, these are used in combinations. Prebiotics include gluco-oligosaccharides, phos, oligofructose, trans-galacto-oligosaccharides, TOS, malto-oligosaccharides, statiose, lactulose, lactitol, sucrose oligosaccharides, xylo-oligosaccharides, and raffinose. Symbiotics. The term symbiotics was introduced in 1995. It is defined as a mixture of probiotics and prebiotics that benefits the host by improving the survival and implantation of live microbial dietary supplements in their gastrointestinal tract (GI tract). It selectively stimulates the growth and/or activates the metabolism of one or limited number of health-promoting bacteria and thus improves the host's health. Its main function is to improve the survival of probiotics in the host's GI tract. Probiotics beneficially constitute a protective barrier for the alimentary tract, and prebiotics at the same time provide nutrients and energy for probiotic bacteria. For selecting a symbiotic combination, the probiotics and prebiotics should meet all the criteria of selection criteria of probiotics and selection criteria of prebiotics. The process of determining the combination of both is difficult and requires a number of studies. A good combination should have a beneficial effect on the growth of selected microorganism with limited or no effect on the growth of rest of the microbes. Prebiotic commercial name combination. Lactose CS 1000 polysaccharides, oligosaccharides DOLSORBDN, Dalphos MOS, cis-glucans. Microstop extrabit MOS, cis-glucans table 12.3 prebiotic combinations. Symbiotic. Commercial name. Prebiotic probiotic. Digestate pediococcus acidilactoside, Saccharomyces cerevisiae, boulardii. Cis-glucan, monooligosaccharide. Symbiotic poultry betafarm. Lactobacillus, acidophilus, casei, salivarius, plantarum, rhamnosus, brevis. Bifidobacterium, bifidum, lactis, streptococcus thermophilus. Inulin. Table 12.4 difference between symbiotic, prebiotic, and probiotic materials. Figure 12.21 microbes in animal health improvement. These have been categorized into probiotics, prebiotics, and symbiotics which altogether help in improving the digestion of rumen, promoting survival of healthy bacteria leading to a better yield of dairy products. Rumen microbiome. The rumen of an herbivore is one of the most complex, richest, and most productive microbial habitats on Earth. It is known as the most effective system for conversion of plant biomass into protein, short-chain amino acids, and gases like carbon dioxide, methane, and hydrogen. It is a rich source of enzymes and genetic material which can be used for industrial purpose and biofuel production. Microbes present in the gastrointestinal tract of rumen help in the digestion of plant linocellulose. They convert carbohydrates to volatile fatty acids, VFA, the main energy source for cow and the main source of fat for milk. They also convert non-protein nitrogen to high-quality microbial protein, used by the cow and also metabolize some plant toxins. Rumen microbial flora is extremely diverse until date only 5% of it has been studied. It contains representatives of all three domains, i.e., bacteria, archaea, and eukarya. Rumen bacteria. Bacteria are most predominant microorganisms in the rumen gut. These occupy up to 10-11 cells per gram of rumen content. These have high fibrinolytic activity and help in the degradation of plant fibers. These microorganisms are found to act on cellulose, pectins, sugars, proteins, etc. Much of the digestion in rumen starts by action of bacteria. These bacteria mainly belong to four phyla: Bacteroidetes, Firmicutes, Proteobacteria, and Spirochetes. Study of 16S RNA has helped in understanding the vast diversity of bacteria occupying the gut of rumen. Majority of bacteria found in the rumen gut include S. No species stain functions. 1. Fibrobacter succinogenes. Gram-negative ferments glucose and produces acetate and succinate as waste products. 2. Ruminococcus flagellations. Gram-positive cellulitic, i.e., helps in plant cell wall digestion because of its high cellulase and hemicellulase activity. It produces hydrogen, acetate, and succinate as end products. 3. Megasphaera alcini. Gram-negative found mainly in young ones and it ferments glucose to propionate making it available for gluconeogenesis. 4. Selenomonas ruminantium. Gram-negative ferments glucose and it occurs in large numbers, especially when ruminants are fed on grain. 5. Phylonella. Gram-negative uses lactate as a substrate and produces acetate and propionate. 6. Butyrolvibrio fibrosolvens. Gram-negative is a cellulolytic bacterium, common in the rumen, and produces acetate and butyrate by fermentation of glucose and other substrates. 7. Lactobacillus ruminis. It is majorly found in the reticulorum of young animals. It uses glucose as substrate and ferments it to produce mainly lactate. Table 12.5 list of important bacteria present in rumen gut. Rumen archaea methanogens. Archaea are prokaryotic organisms which produce methane as the end product of the biochemical process. Rumen guts are an impressive host of a diversity of methanogens. Methanobacterium ruminantium and Methanosarcina barbarii are imported among these. These are anaerobic and exist in an anoxic environment. These are found to exist in the GI tract of animals and humans. They produce methane as an end product which is a major greenhouse gas. Almost 29% of total anthropogenic methane is produced by ruminants yearly. Therefore, understanding the process of methane production and control of its production might be very helpful. Phage therapy has been found to be instrumental in the eradication of microorganisms and controlling their population. This can help eradicate methanogens from the gut of rumens as well. Venturing into this field might be helpful in future to control level of methane emission of rumens. Eukaryotic protozoa and fungus diversity in rumen. Rumen protozoa occur widely, i.e., 104,106 per gram of rumen. These were discovered by Gruby and colleagues in 1843. These ciliates are identified mainly on the basis of microscopic observation. At present, 18S gene sequence along with microscopic observation is used as a molecular marker for their phylogenetic characterization. There is a lack of reference sequences related to their architecture. Therefore, their metagenomic study is still in disrupting the cell wall of foodstuff and making it available to biodegrading rumen bacteria. Fungi account for approximately 20% of the total rumen microbial biomass. These fungi are highly oxygen and temperature sensitive, which makes them difficult to grow in vitro. These possess very high E+T content. Rumen inhabiting anaerobic fungi were first described in 1975 by Orban. These are said to lack mitochondria and possess hydrogenosomes, which ferment pyruvate and malate and produce hydrogen and carbon dioxide. Approximately 17 different fungi which belong to 5 different genera have been reported in 50 different herbivores. The most common prevalent anaerobic fungi in cattle include Aspergillus niger, Neoplomastix frontalis, Enderliensis, Orchinomyces bovis, Ruminomyces elegans, and Armucronitis. These anaerobic fungi are difficult to grow in culture. Special tubes and serum bottles sealed with stoppers are required to grow these in small batches. Different media compositions have been suggested to grow different strains of fungi. Infancy. About 124 protozoa species have been observed in the enriched sample as compared to just 29 protozoa species in the whole rumen sample. Out of these, Entominium, approximately 28%, is the most abundant species, followed by Toxoplasma and Epidenium. Their population in rumen is very large, with a mass approximately equal to that of bacteria. These are supposed to play an important role in hydrolysis of carbohydrates, proteins, and lipids. The homology between Entominium pallidum and the closest non-protozoan homolog gram-positive bacterium indicates that horizontal gene transfer leads to the acquisition of protozoans, cellulolytic genes from bacteria. Some examples of protozoans include Isotryptoprostoma, I. intestinalis, Epidenium pallidum, Entominium bovis, E. bursa, Eutypolonium magi, Diplodonium dendatum, etc. Fungus is known to represent around 10 free spores per milliliter of rumen content. It also plays a very important role in utilization of food as it mainly contains fibrolytic enzymes. Fungi are the first ones to help in digestion by Figure 12.22 Rumen microbiome. Unexplored. These species were never cultured before and hence indicated the microbial community is far more complex than known by culturing methods. Metagenomics has two aspects, which are structural and functional metagenomics. In the structural metagenomics approach, the aim is to figure out the structure of uncultured microbial population in the sample. The information on structural composition of the microbial population may lead to a metabolic network established between the members and dynamics of the population in response to certain selection pressure or spatiotemporal parameters. Structural studies help in deciphering the relationships between the community members which are essential for biological and ecological functioning of the community. On the other hand, functional metagenomics approach targets identification of genes with desired functions. This involves the generation of expression libraries with metagenomic clones which are screened by activity-based assays. Fig 12.23. IT is known as the most effective system for conversion of plant biomass into protein, short-chain amino acids, and gases like carbon dioxide, methane, and hydrogen. It is rich in bacteria, methanogens, protozoa, and yeast which help in digestion of complex food and fibers using a variety of enzymes. Microbial metagenomics. The term metagenomics means more than genetic information. The study of genetic material recovered from environmental samples or biological samples is called metagenomics. The term metagenomics was coined in 1998 by Joe Handelsman, John Clardy, Robert M. Goodman, and Sean F. Brady, who used it first in the publication, where it meant genes collected directly from environmental samples can be analyzed in a similar way as genome of a single organism. But before this, in early 1986, Norman R. Case used PCR to study the diversity of ribosomal RNA in environmental samples. His study led to the existence of a complex community of microbial populations, which was Figure 12.23 schematic arrangement showing metagenomics approaches. 3. Next generation sequencing, NGS, derived shotgun metagenomic studies. This is a high throughput format of sequencing. Various sequencing methods like 454 pyro sequencing, Luminet selective sequencing, solid sequencing by oligonucleotide ligation, and detection massively parallel signature sequencing, MPSS, choline sequencing, ion torrent semiconductor sequencing are available for metagenomic studies. These technologies are capable of delivering massive amount of genomics data, which then requires expert bioinformatics tools and personnel to be analyzed. NGS offers an advantage of providing secondary information about the genomic sample like evolutionary relations, species identification, prediction of unique metabolic pathways, and prediction of resistance. Metagenomics approaches that reveal human microbiome over time. Revealed microbes are not only responsible for various infections and diseases, but they also play key roles in health and metabolism. For example, human gut microbiota is composed of 10 microorganisms which include bacteria, fungi, protozoa, and viruses. The metagenomic studies by advanced NGS technology and omics techniques have revealed that gut microbiome synthesizes essential amino acids and processing of cellulosic material which is important for health. Also, microbiome protects against the pathogenic strains of bacteria and protozoa. Metagenomic studies using stool samples from colorectal samples have been used to develop biomarkers for colorectal cancer, which is a non-invasive method for early diagnosis. Biotechnology has immensely benefited from metagenomics. As the microbial world has always been a major source of commercially required biocatalysts, and metagenomic studies are useful in mining out genes from the environmental pool. One of the major challenges in metagenomic studies lies in the functional screening of the desired genes from the metagenomics library. The robust high-throughput format of screening methods needs to be developed to keep up the environmental samples can be surveyed for biotechnological features, which form functional metagenomics side. While studying the structural features of the environmental DNA sample will reveal the constituting microbial flora. The study of 16 srRNA may actually help in classifying the microbial flora and understanding the web of the particular niche. It should be noted that 16 srRNA surveys are sometimes considered as metagenomics, but since it is a study based on a single gene, is not more than a molecular taxonomic study. Nevertheless, approaches which establish links between 16 srRNA and genes or metabolic pathways are useful in determining the functional potential of microbiome. The metagenomic studies can be divided into following categories on the basis of methodological approaches adopted. 1. Foch-Mead, Cosmet, and bacterial artificial chromosome (BAC) derived metagenomic studies. In 1999, Rondon et al. First produced BAC library of NGT, 10 base pairs by DNA isolated from soil sample. Later on, in 2002, Beige et al. Created a Phosmet library from waters of Antarctic and deep Pacific Ocean. Phosmet-based libraries become important in the study of marine microbiomes due to their technical advantages. The most important studies carried out by Phosmet libraries have been in 2006 at Hawaii Ocean Time Series Station in the Pacific. The data generated through the study revealed that certain proteins and clusters of contiguous proteins were present at specific depths. This was equivalent to the species summation on land. 2. Sanger sequencing, derived shotgun metagenomic studies. The sequencing of small insert libraries by traditional Sanger sequencing is also a useful approach to studying microbial metagenomes. In 2004, this technique generated 1 billion non-redundant base pair sequences from oceanic waters from the Sargasso Sea. These sequences contained 1,800 unique genomes, 48 unknown bacterial phyllotypes, and 1.2 million new genes. Type of error: the high-frequency barcodes, or indices, that are designed to uniquely identify multiplex samples can be wrongly assigned during scanning of the flow cell. This may lead to false positive detection and exaggerated readouts. But the latest platform, NovaSeq, redesigned with pattern flow cell in which the exclusion amplification process maximizes the number of non-holes occupied with a cluster of sequences originating from single template, and hence increasing the quality of data output. NovaSeq can deliver up to 1.6 billion reads in 2-lane flow cells with greater than 85% accuracy. Another platform from Thermo Fisher Scientific is called Ion Torrent. In this platform, the DNA is amplified on a bead in an emulsion. The beads are then placed on matrix of highly sensitive pH sensors which detect the local changes in pH which occur during sequencing by synthesis and identify the sequence nucleotide. This chemistry is also used in Roche 454 sequencing platform. Another platform from Life Technologies is called solid sequencing by oligonucleotide ligation and detection is available, which also uses emulsion PCR technique which occurs in a microreactor environment. The beads are then covalently linked to the glass surface and hybridized with primers complementary to the adapter sequence in the library template. For sequencing, 4 sets of D-based primers are made which are fluorescently tagged. These primers compete for the ligating to sequencing primer. With these primers, multiple cycles of ligation, detection, and cleavage are performed for sequencing. The specificity of D-base probe is checked by keeping track of first and second base of every ligation reaction. According to manufacturers, this platform can provide up to 60 gigabases of usable DNA sequence data per run with 99.94% accuracy. Schematic presentations of various NGS platforms are described in Figure 12.24 for visualization of the overall process. Pace with the amount of metagenomics data which is being produced. Hence, by combining collective efforts and new approaches will help in diving deeper into the unseen world of microbes and harness the untapped potential lying there. Overview of NGS and platforms used in the technology. NGS is also known as high throughput or massive parallel sequencing. This genus of technology has been developed for sequencing thousands to billions of bases simultaneously and independently. The application of NGS to metagenomic samples has been immensely appreciated by the scientists. This technology provides an unbiased approach to identifying the microbes present in an environmental, clinical, or ecological niche sample through mass sequencing. Apart from environmental samples, NGS can be used for clinical samples for assessment of infectious diseases, lineage tracing, drug resistance testing of viral or culture samples. The machines used for such massive sequencing are called platforms. There are various platforms available that have unique chemistry for sequencing. The methods are standardized and commercially available. One of the most popular platforms is offered by Illumina, San Diego, California. The various versions of platforms are named as iSeq, MiSeq, MiniSeq, NextSeq, HiSeq, and NovaSeq. All the platforms use common chemistry involving the bridge amplification process. In this process, single molecules of DNA fragments are attached to a flow cell and then amplified into a local clonal cluster. Now the clonal DNA cluster is sequenced by a synthesis cycle, in which a complementary DNA is built and an optical signal of fluorescently labeled nucleotide is then determined. As of now, Illumina has the highest throughput of all sequencing platforms, but it suffers from the problem of barcode index switching. In this Figure 12.24 schematic representation of workflow of most commonly used NGS platforms. The process is divided in 3 steps: library preparation, library amplification, and sequencing. All kinds of DNA samples are converted to double-stranded short DNA fragments which are flanked by known sequences called adapters. These end sequences are specific for a platform and enable the binding of library molecules to the surface of either flow cell or beads, where clonal amplification of the library molecules takes place prior to sequencing. The clonal population of amplicons are spatially segregated on glass slides and chips or pipetide or plate. Amplicons can be sequenced by various techniques like formation of ice crystals which leads to cellular injury. The fast cooling of cells leads to ice crystal formation while slow cooling leads to osmotic shock. The effects of freezing on a cell can be altered in presence of a cryoprotective agent, CPA, or also called cryoprotectant. These agents alter the rate of water transport, nucleation, and ice crystal growth. There are organisms that can tolerate freezing and have natural CPA molecules. Tardigrade water bear are microscopic multicellular organisms that can tolerate freezing by replacing all internal water with sugar hollows. Also, one of the frog species Rhinocellatica uses glucose as a cryoprotectant when cooled slowly. Cryopreservation of tissues is different than single-cell suspensions and is very challenging. The transfer of heat and mass distribution uniformly is difficult to achieve in bulk tissues along with uniform CPA distribution. Although the task of cryopreserving the organs, tissues, and biological material is challenging, but the immense clinical applications and basic research potential is motivating researchers to develop techniques for effective preservation. The successful cryopreservation is now getting closer to reality through the discovery of CPAs and temperature control machines. The procedures and agents involved in cryopreservation vary according to the cell type and can be of following type. 1. Slow freezing. 2. Vitrification, which is a process of converting the aqueous milieu of cell into non-crystalline glassy phase. 3. Sub-zero, non-freezing storage. 4. Preservation in dry state is not very feasible as introducing CPAs in intracellular regions is very difficult. The cryopreservation protocol mostly follows the below-listed steps. 1. The CPAs are mixed with cells or tissues. 2. Cooling of cells to lower temperature, maybe in steps, and storage. 3. Thawing of frozen cells or tissues. 4. Removal of CPAs from the cells or tissue and providing those suitable conditions to grow. Sequencing by synthesis is in case of Illumina sequencing by ligation process with labeled oligonucleotides solid. But in case of ion torrent sequencing, the unlabeled nucleotides are flushed over the flow cell in sequential order and incorporation is detected via proton release and shift in pH. BGI SEQ platform, which is offered by BGI, Cambridge, Massachusetts, again, uses amplification of DNA molecules in a flow cell which produces DNA nanoballs. These nanoballs undergo sequencing by synthesis. The sequencing is carried out by synthesis and the readout is processed in a fashion similar to the Illumina platform. This platform is yet to be commercially available but is used for sequencing infectious disease samples from clinics. Lesser used NGS platform from Oxford Nanopore Technologies are named as Minion, Viridian, and Promethin. This technology uses gridded protein nanopores that gather single-stranded DNA through electrical pulses. This sequencing is free from PCR amplification of parent DNA molecule and sequences of fragments through synthesis. This technique is useful for infectious disease diagnostics as it is faster than other methods by several orders. Since this technique is PCR free, there is a requirement for high DNA input of greater than 500 nanograms. However, comparison of this technology with counterparts reveals that it has more sequencing errors and lower throughputs while the cost of each read is higher than other NGS platforms, which may limit the commercial utility of this platform. Cryopreservation. Cryopreservation is a method of preserving cells or tissues in a state of suspended metabolic activities at very low temperatures. Theoretically, cryopreservation aims at maintaining the viability of cells upon indefinite time of preservation followed by eventual thawing. At very low temperatures, the enzymatic reaction and other chemical reactions can be practically stopped, which is required for long-term preservation of cells and tissues. But freezing is not tolerated well by most organisms, cells, and tissues because of the University of California. His body continues to be preserved in liquid nitrogen at the Alcor Life Extension Foundation. Every year, January 12th is marked as James Bedford Day, as he was cryopreserved on this day. Polymers. The vinyl-derived polymers like polyethylene glycol C2NH4N plus 2 on plus 1, molecular weight 200-9500 daltons, polyvinyl alcohol C2H4ON, molecular weight 30 to 70 kDa, and hydroxyethyl starch 130 to 200 kDa can encapsulate the cells during suspension and reduce the formation of ice crystals. Cell Banker series. This cryopreservation media from Nippon Zenku Kogyo Co. Limited has been shown to achieve better survival rates. This CPA contains 10% DMSO, glucose, and high polymer and pH adjusters. The serum contains series is ideal for preserving most mammalian cells. While non-serum series is composed of all known ingredients and is chemically defined and is suitable for cryopreservation of stem, somatic, and induced pluripotent stem cells. Methods of freezing. Cryopreservation is carried out by slow freezing and vitrification process. The two processes differ in the CPA used and the rate of cooling applied. It is proposed that, if cells or tissues are cooled slowly, the intracellular excess water is effluxed out rapidly to prevent supercooling and hence the formation of ice crystals. But different cell types vary in their capacity to move water across the membrane, so the rate of cooling is required to be optimized for membrane permeability. Slow freezing technique helps in substituting intracellular water with CPA, which reduces the cryoinjury to the cells. Most commonly used cooling rate of 1C is applied with a controlled freezer in presence of not more than 0.1 m of CPA. Slow freezing method comes with benefits like low risk of contamination, but it has a high risk of cryoinjury due to the formation of extracellular ice. Vitrification is an alternative to the slow CPAs. These agents are usually fluids that reduce the injury due to the cryopreservation process. An ideal CPA should have low toxicity, be biologically tolerable, and should penetrate the cells. There are many CPAs that reduce the formation of ice crystals at any temperature, depending on the cell type, freezing, and thawing rates. To achieve the best survival rates after thawing, the CPA concentrations, volumes, cooling rates have to be optimized for the cell or tissue under investigation. The cryoprotective agents are classified into two types. 1. Cell-permeable CPA such as dimethyl sulfoxide, DMSO, and glycerol. 2. Non-membrane penetrating CPA such as 2-methyl-2, 4-pentanediol, and polymers such as polyvinyl chlorolidone and various sugars. Biomaterials such as chitins and alginates can also be used as cryoprotectants along with small molecules to impede the growth of ice crystals during freezing and thawing cycles. Glycerol. Glycerol is a cryoprotectant was discovered in 1949 by Kolch et al. This is a non-electrolyte polyol that acts by reducing the electrolyte concentration in residual and frozen solution inside and surrounding the cells. It is widely used in cryopreservation of bacterial cells in research laboratories and sperm cells. DMSO. After glycerol, DMSO was demonstrated to be an effective cryoprotectant by Lovelock and Bishop in 1959. It is also a non-electrolyte and works by reducing the electrolyte concentration in an unfrozen solution. Since DMSO is inexpensive and has low toxicity for mammalian cells, it is widely used as CPA for cultured mammalian cells. It has been also shown to induce cell differentiation by DNA methylation, so its clinical applications are limited. But still, DMSO was the main cryoprotectant used for preserving the first human, James Hiram Bedford, who was a psychology professor at Cells, like cancer and its treatment. Moreover, cryopreservation of embryo is also performed and has been shown to preserve the viability for long terms without any effect on subsequent pregnancy. Cryosurgery. This is an application of cryosurgery to cells and destroys them without having any toxic effect on the organism. By this method, external tumors, such as on skin or any abnormal tissue, are destroyed by focused freezing with liquid nitrogen. The advances in cryosurgery have dramatically reduced the side effects associated with the treatment of the disease. Fig 12.25 describes the effects of freezing during treatment and preservation of cells and tissues. Figure 12.25 affects an application of freeze thawing. The survival of cells after cryopreservation depends on parameters like freezing temperatures, cooling rates, and cryoprotective additives. For cryosurgery, the harmful effect of freezing is utilized to destroy the malignant cells, while in the case of preserving the cells and tissues, the effects of cryoinjury are tried to be minimized. Rapid cooling is known to cause formation of intracellular ice crystals (IIC), which damage the cell during thawing. On the other hand, slow cooling causes EFF, flux of water, and dehydration of cells. The protocol needs to be optimized for harnessing the advantage of damaging effects and therapeutic effects of freezing. Freezing method in which the cell suspension is directly transformed from liquid phase to glass state by direct exposure to liquid nitrogen. The process involves high concentrations of CPAs, which may range from 40 to 60% weight volume. In presence of high concentrations of CPAs, the rapid cooling results in the transformation to a glass state avoiding ice formation. Vitrification process largely depends on three factors. 1. Viscosity of sample. 2. Rate of cooling. 3. Volume and mass of the sample. Furthermore, the vitrification process is categorized in two types, equilibrium and non-equilibrium. In the first type, the multimolar CPA formulation is included in the cell suspensions. In case of non-equilibrium, there is a carrier-based method which involves use of loops and microcapillaries to obtain accurate volumes of sample. While in the case of carrier-free systems, the samples with low concentration of CPA are frozen at very high freezing rate. The vitrification offers the benefit of less risk of cryoinjury, but it is also accompanied by risk of pathogenic contamination of samples. Applications of cryopreservation of cells and organ stem cells are potential gold mines for regenerative medicine, tissue engineering, and gene therapy. These cells are divided into embryonic, mesenchymal, and stromal cells. Pluripotent stem cells are capable of differentiating into multiple types of specific terminal cell types. These cells can be obtained from cord blood and can differentiate in any cell type except for placental cells. The cryopreservation of these cells is essential for the timely supply of viable stem cells for medical treatment. Another application of cryopreservation is in reproductive medicine. The cryopreservation of mature oocytes is now a known method of preserving reproductive potential. Oocytes are large cells and more prone to cryoinjury, so preservation of immature oocytes and ovarian tissue is a promising approach. Similarly, sperms and semen are also cryopreserved to ensure patients with diseases whose outcome may affect germ errors, and soma clonal variation requires cost-effective cryopreservation methods. Cryopreservation by liquid nitrogen is a safe and cost-effective technique for germplasm conservation of non-orthodox seed species, vegetative propagating plants, and other biotechnology products. New cryopreservation techniques are based on vitrification by internal solutes and are successfully used with almost all X plant types, including ecdyses, somatic or zygotic embryos, and callus or cell suspension of temperate and tropical species. The cryopreservation methods are more sophisticated for vegetative propagated species. However, the technology is also used for different types of material, including dormant buds, pollen, and ecdysis samples from vegetative lab propagated species. Cryopreservation is also used for cryotherapy, in which the viruses are eliminated from the plants through apex preservation. Because of the multiple applications of cryopreservation methods, it will be highly used in future in plant biotechnology. Limitations. With numerous advantages of cryopreservation techniques in basic and clinical research, some limitations still exist. The metabolic and active state at low temperatures can cause genetic drift or may result in cell-associated changes in proteins or lipid which may impair cellular structure and functions. The CPAs used during the preservation may result in damage to the cells like in case of DMSO, which may lead to DNA methylation and altered genetic stability and might lead to oncogenic variation. Lastly, the cryopreservation methods involving manual manipulation may lead to contamination with pathogenic agents which is not desired in clinical and scientific procedures. Development of transgenic animals before the dawn of molecular biology and genetics. The only method to study the function and regulation of genes was by observing inherited characteristics or spontaneous mutation. Method of selective breeding 2. Biochemistry and molecular biology. Biological research many times depends on the cells or clinical samples preserved before. The preservation must be effective in preserving the biochemical status and cellular features of the samples. Cryopreservation is very essential in this aspect. Apart from this, the preservation of bacterial strains in glycerol for long periods is very common. Similarly, preservation of viral particles by freeze-drying method is another approach. Preservation of viral particles is essential for experimental research. Production of vaccines and alteration of the genome by viral-mediated methods is in the case of gene therapy. Food sciences. Meats and agri-food products are important for sustaining populations. Many agri-products like vegetables, fruits, and meat are highly perishable. Their distribution across the population and development of industry depends on the effective preservation of the quality. Since these food products are high in moisture and nutrients, they form an ambient niche for pathogenic organisms to flourish, leading to deterioration in quality. The traditional freezing techniques like air blast freezing, cryogenic freezing, and plate contact freezing are slow freezing processes which lead to the formation of large irregular crystals of ice, which damage the meat and fish products and some agri-food products also. Now novel freezing methods like high-pressure freezing (HPF), electrically and magnetically assisted freezing (EF and MF), ultrasound-assisted freezing (UAF), and antifreeze coating (AFP) have been developed for preserving the food. The technology involves the application of an electric or magnetic field during freezing which can affect the polar molecule like water and can control the ice crystal formation. Plant physiology. Cryopreservation ensures the genetic integrity and viability of the plant material for long term. The maintenance of cell lines and minimizing the risk of losing to contamination. Technical possibility. The foreign injected DNA may also cause over or underexpression of certain other unrelated genes as this technique is not target specific. The microinjected ovum is then transferred to the oviduct of the recipient female as described in figure 12.26. The advantage of this method is its applicability to a wide range of species. The probability of successfully producing transgenic mice by pronuclear microinjection varies with the DNA constructs, with an average rate of 30% live-born animals. Sometimes efficiency of 90% has been reported in the case of plasmid PBR322 digested with PSTI enzyme before microinjection. The insertion of the foreign gene after microinjection is poorly understood, but Costantini and Lacey et al. Have reported head-to-tail insertion in multiple copies in the genome. The number of copies inserted in the genome cannot be controlled, but insertion leads to rearrangement of host genomic DNA-like translocations. The inserted gene may also lead to the inactivation of the normal gene by insertional inactivation. The resulting disruption might lead to an oncogenic phenotype. Post-insertion, the gene of interest needs to express, but the level of expression is controlled by many factors. For example, beta-globin gene which expresses in adult bone marrow is sequentially activated during the development. The initial experiments involving the full gene of beta-globin inserted in transgenic mice resulted in no expression. But later when smaller fragments of the gene were microinjected, it resulted in an expression which was far below the endogenous expression levels. Later it was found that the expression of this gene was regulated by sequences 50 kilobases far from the gene, and co-transfection of that sequence with full beta-globin gene resulted in expression comparable to the copy number of the gene present. Increase in milk and agricultural yields has been practiced long before the Mendelian discovery of genes and was method of selective enhancement of traits. Discovery of DNA and its role as genetic material has been a breakthrough in molecular biology. Since then, there has been tremendous growth in molecular biology leading to the development of techniques by which we can alter the genome of an organism. Transgenic animals are organisms in which the genome has been modified experimentally in contrast to spontaneous mutations. In short, the DNA sequence coding for the desired characteristic is introduced into the organism stably, which should then be transmitted to all the cells of the organism. For reasons of easy availability and maintenance in laboratory conditions and shorter lifespans, mice are the most appropriate animals for transgenic research. The first chimeric mice were developed in the early 1970s. The cells of two different embryos at each cell stage combined to form a single embryo which subsequently developed into a chimeric adult. The advancement in both developmental biology and genetic engineering allowed the development of techniques for creating transgenic animals. In 1981, J.W. Gordon and F.H. Ruddle first used the term transgenic, and since then, there has been unprecedented growth in the field. There are three methods by which transgenic animals can be created. DNA microinjection. This technique involves direct injection of selected gene construct from the same species or different species into the pronucleus of a fertilized ovum. It has been one of the first methods which proved to be effective in mammals. The inserted gene may or may not express itself with a high probability of later figure 12.26 DNA microinjection technique used for producing transgenic mice. In the procedure, female mice are superovulated and mated, and a pronucleus of the fertilized egg is microinjected with DNA top right. The resulting surviving zygotes are reimplanted center in the surrogate mothers, and newborn pups are tested by Southern blind for incorporation of new genes bottom. Coat color markers are used to identify microinjected embryos for testing the presence of the foreign gene. Embryonic cell-mediated gene transfer. In this method, DNA is inserted into the genome of embryonic stem cells by homologous recombination method and can be considered as a nearly perfect method of gene transfer. Stem cells can differentiate into any type of cell, somatic or germ, and hence can give rise to any organ or whole organism. These genetically modified or edited cells are then mixed with the blastocyst stage of development, which results in the chimeric organism. The S-mediated gene transfer is the preferred method for gene inactivation or knockout method. This method has the advantage of performing targeted gene editing in the genome via homologous recombination and is carried out as shown in figure 12.27. But this kind of gene insertion is rare and the selection of recombined cells becomes challenging. The most common tool for selection is neomycin resistance, which is gained when donor DNA is applied to S cells, and modified cells can be identified by resistance to neomycin. Additional markers like herpes limiting kinase can be used, which gives resistance to the antiviral drug ganciclovir. The double selection makes probability of selecting cells that have undergone homologous recombination. As this method is mostly used for knocking out the expression of genes, it provides deeper insights into the role of a particular gene in developmental stages. As reported by Masensky et al., the deletion of CMYB results in failure of shifting the site of erythropoiesis in the fetus from yolk sac to liver. Figure 12.27 embryonic stem cell mediated production of transgenic mice embryonic stem cells are cultured and subjected to DNA-mediated gene transfer (DMGT), top or other manipulations. Cells with the desired genetic alteration are then inserted into blastocyst cavities of the embryo. When these embryos resume normal course of development, a genetically mosaic mouse is produced in which some cells are derived from the S line and the others from the injected blastocyst center. Founders, usually males, are then bred to pass on the gene, provided the S cells differentiate into sperm. The F1 hybrid then carries the new gene in all cells bottom. Retrovirus-mediated gene transfer. To increase the efficiency of gene transfer, a carrier or vector which is generally a virus is used. Retrovirus